Welcome to the 2021 California Leafy Greens Research Program Annual Research Conference. How you can participate today, it will be recorded and distributed probably sometime next week. Once Jennifer and I get it coordinated across and she gets it up online, we'll send you just an email letting you know it's there. For now, type your questions into the Zoom's Q&A option. It should be at bottom of your Zoom ribbon over here. Make sure to put the speaker's name before the question so we know who you are asking. Don't use the chat for questions, use it for comments only. Make sure you use this little drop down item to make sure it's going to all panelists and attendees. That way everyone can see your chat. But again, put the questions in Q&A and save the chat for comments and discussion. It's just easier to keep track of that way. For continuing education, if you were attending yesterday, a test was already sent out. So if you didn't receive it, you can send me an email. I'll make sure I get it to you. But for today, we have 3 hours in the other category. Make sure to answer the live poll questions. If you can't, for some reason, your pop-up's not working, just type your answers into the chat. You got to be present for the entire event and then take the test that will be emailed to you after. You have 1 week to take the exam and only 1 user per device can get credit. So make sure if you have several people listening to one computer, you call in so that each person can get credit. Certified crop advisor, same thing as yesterday. We have an assortment of credits and a QR code will be put up at the end of the meeting. Once that QR code is up, you can scan it with the app and get your credit automatically. No worries. If you don't have it, just send me an email with your information. And that is it. I'm going to turn over to Jennifer.
So welcome everyone to our day 2. I am gonna just rehash a little bit of the slides that I kind of went over yesterday, but had a little technical difficulty. So I want to ask, can you see my slides?
Yes, looks great.
Perfect, thank you. So this too is just, I've left this up just so y'all can see who represents our Leafy Greens Research Board currently. This is also on the website. We talked a little bit yesterday about industry consolidation, and I'm sure many of you are quite aware of this. So, back in 2013, we had 94 handlers participating in this program. And because of consolidation, we're now down to 67. Here's our lettuce volume. So we've got iceberg, romaine, leaf lettuce. Try this little laser pointer so you can see the different commodities. And probably not surprising to everyone, with COVID and INSV, Pythium type issues, we really had a double whammy of a year, and we saw some volume decrease. Here's our spinach and spring mix volumes and trends. We saw a little bit of a decrease in a couple of these categories and an increase in one. And I can share these slides with anyone who'd like them. Here again is just what we're funding. This is for next year, how it breaks down for our next fiscal year, which really starts April 1st. So these are the projects categories that we'll be funding. And I do have this picture up behind me just to really demonstrate the kind of devastation that we saw with INSV and Pythium last year in Salinas. So while we're going to hear a little bit about INSV kind of at the end of today, we definitely next year, you're going to hear a lot more about it because we're funding more projects related to that and Pythium as well. So again, here's the challenges. They sort of remain the same, although, we have different things that will kind of come up, which honestly is why then it's so important to have our farm advisors and specialists. And we know that UC-A&R, maybe not all of you realize, but UC-A&R is currently in a hiring freeze due to budget constraints. And with that, there's my contact info. You can find it on the website as well, but feel free, and I mentioned this yesterday, even if you're not participating on the board, which the board really helps set the priorities and our research funding, feel free to call me or email me anytime if you've got questions or concerns, or you're really interested in finding a solution to a problem. I'm always open to taking those suggestions. And so with that, I'm going to stop sharing my screen. And there we go. And we can get started, and it'll be great if we stay on schedule or ahead of schedule. So Nathan, if you want to share your screen. Nathan Ju is our first presenter today. Perfect. We can see, that looks good.
Great. Do you want me to wait until 9:10 or just get started?
Nah, get started. It'll be
Okay.
awesome to be ahead of schedule.
Perfect. Well, so thanks for having me here to talk about our project. And obviously thanks to Leafy Greens for funding this project, which you guys have done for the past 2 years. So this year, we had sort of progressed our work on the bioreactor system that John Silvis and Arlene Hoffett originally developed, and continued on to do what we had hoped to do was some field optimization of water flow. So we get a better idea how to scale up these bioreactor systems. So today I just wanted to give you guys a little bit of an overview, kind of some of the other work that we're doing that supports this, and then how this relates to our current project. So our long-term goal for this project is generally to develop a low-cost, small footprint bioreactor system that's going to leverage naturally occurring microbial communities to decrease the environmental impacts of various agricultural activities related to agricultural chemicals, such as pesticides and nitrogenous wastes. And we have a couple different components of this. One of the things that we've been doing for a while is looking at some genomic objectives, trying to understand, we found these naturally occurring bacteria that can remediate a whole suite of different types of pesticides. We don't really know how they work. So we've been spending a fair amount of time, trying to figure out the mechanisms behind these bacteria and what's sort of driving their ability what's the biology behind how they're able to accomplish this task, in the hopes that we can use that information to design a better bioreactor system. And so we've been doing a bunch of genomic work, trying to figure out, what are the mechanisms, what's the make of these things, what are these things, which ones of them remediate better than other ones, which ones have more phenotypic capacity to eat pesticides, basically. And we found about a bunch of different strains for various different targets. A lot of these targets we started off with, they look probably a little bit old compared to what some people might be using these days, but these were originally targets that were emphasized to us by the CDPR, California Pesticide Regulation. And so that was one of the reasons why we focused on these. But there's a suite of pyrethroids and neonics that would be interesting to folks. And so we've been able to identify a bunch of these genuses. We've got pretty good reference genome sequences for these things. So we can start doing a lot of really interesting experimentation on them. And we found some cool things in the process where we found like some of these bacteria have acquired certain genes that are related to remediating pesticides, like this P450 gene for this imidacloprid remediating taxon. We find 2 copies of it that we don't find in its closest sequenced relative. So it looks like a gene that it's acquired to help it with the remediation of imidacloprid. Whereas we might look at another imidacloprid remediating organism, one that actually does a really good job of breaking down imidacloprid, and we don't see those genes. So we're actually, still looking for novel mechanisms in some of these organisms, trying to figure out what's the process that's driving this. So those are the sort of like lab components that we've been doing to try to develop the system that's actually going to go into the field. Along those same lines, we've had work funded through Leafy Greens to try to take that lab work and implement it out in a field environment, a practical environment. And so we've been doing a couple different things. So the first year, I'm just going to briefly review what we found, and then I'll talk about what we've been working on this past year. We're basically trying to figure out how do these things work. If we put this in the environment, what effect could it have? How much could it possibly reduce pesticides in the field? And is it a feasible, reasonable solution that people might be interested in implementing? And so we constructed a field bioreactor 2 summers ago. We constructed this bioreactor. We attached it to the outflow, the tailwater outflow of a field in Salinas Valley. And then we monitored a bunch of the environmental conditions. And we basically, the first time we just did a test to see, well, what happens if we don't add bacteria? And then what happens if we add bacteria? Do we actually see an effect of these bacteria on the amount of pesticide in the water? So it's an interesting system. It's pretty small in its footprint. It's an insulated bioreactor. It's a wood chip bioreactor, so it's filled with wood chips. It's fed directly from the drainage, the pump drainage system from this field system. And basically water runs in up here and then runs through the bioreactor and comes out the other side. Pretty simple system. Wasn't that expensive to build, a few $1,000s if that. It's been holding up really well, still works just fine a couple years out. So seems to be pretty effective. We ran the first summer, we ran a bunch of different experiments. We ran basically 2 weeks where we didn't have any bacteria in, and then 3 weeks where we did. And then we basically added in about 18 different strains, 18 of those different strains I showed you earlier. And the crops that were in the adjacent fields were head lettuce, romaine, and cauliflower. And there was a bunch of different pesticides applications during that time. And what we were targeting mostly was looking at neonics like imidacloprid, and then pyrethroids like lambda cyhalothrin and permethrin. We basically just set it as high as we could possibly, as fast as we could possibly run water through it. And then measured a whole bunch of data, measured a whole bunch of environmental factors, and looked to see if we could see any effect of adding in these bacteria. And what we found was that, yeah, we did see some effects. So what generally happened was we saw an increase in the diversity of the community after we added in the treatment. And so we're adding in bacteria, so we may be artificially increasing the diversity of the organisms there. But we did see this shift. So if we measured both the bacterial communities in the water and the bacterial communities in the soil, and we kind of see this shift from blue to red, where after we add in the stuff, that looks like that we see this change in the community. And then if we looked at the effect on pesticides, we saw significant effects on the concentrations of imidacloprid. But it was kind of equivocal what we were seeing in terms of differences with pyrethroids. So the pyrethroids were difficult for us to, we may not have been doing a great job of measuring them. We're really sure, but the data was a little bit less clear exactly what was going on. But so we thought that this was promising at the fact we were showing about, like a 25% decrease anywhere. So the pyrethroids were decreasing where we could measure it well, around like 10 to 15% of from one end of the bioreactor to the other, whereas imidacloprids, when we could measure it, get good measurements, they were decreasing. Either we couldn't detect them anymore, or they were dropping about 25% from the beginning of the bioreactor to the end of it. So that led us to this next experiment that we were funded to do this past year, which was to look at the existing bioreactor system and seeing if we could get a better sense of water residency time. So how much of an effect, would changing the amount of time we leave the water in the bioreactor, if we slow the flow down or if we speed it up, how would that affect the bioreactor's ability to reduce pesticides? And we kind of did some things to make it a little bit more efficient for us from a sampling perspective. But we're basically going to run at 3 speeds, kind of slow, sort of mid speed, and then fast. And the idea was that if we could get a sense of how quickly the water needed to move through there, we could start to get a sense of how much bioreactor you need to actually service a certain amount of water to a certain, and to target a certain reduction level of reduction. So the way that this was set up was we were going to do some experiments looking at water residency. So we were going to do some detailed tests to see how long it took for plumes of dye to move through the bioreactor. So we get a very accurate assessment of what the actual speeds that we were measuring here at this intake flow meter, how they related to how quickly things were moving across the bioreactor, as well as testing efficiency at different speeds. So we're going to test at these 3 different speeds, these 3 different flow rates, and measure all the things we were looking at. And again, the same types of pesticides were being applied to these adjacent fields. So we expected to be have similar, our methods that had similarly worked pretty well would work well in this case also. We have this is what it looked like before we did before the pandemic happened. You know, we had lots of space and we could do lots of things. But in March, when everything happened, we ended up with a whole suite of other challenges. And you know, probably the most sufficient is that our university shut down completely until the end of June. I was then able to restart research, but really I couldn't restart until August because I couldn't get any of my staff back into the region. And even then we were under all kinds of other restrictions due to fire and smoke. And we had positive tests for COVID-19 in the lab that shut us down for periods of time. And then at various points, the university, we were having to drop our lab densities and field worker densities down to below 25% of what we would normally do. So it became pretty challenging for us to get a lot of this work done. So, you know, we didn't quite get done everything we had hoped to get done in that period of time. And so we were able to run, we had originally proposed to do 9 weeks of sampling. We kind of got done about like 4.5. So we still have some work left to do this summer. We were, because of the late start, we hit right at the end of the growing season in August and September, and we even sampled into October, but we weren't able to get everything in that we needed to get in. So we kind of got messed up from all the delays. But we've got the staff in place now. So we're gonna, our plan is to finish doing some of these water residency tests that we still need to do, and to finish up the remainder of our sampling this spring and summer. But we saw some really interesting data in the weeks that we were able to sample. So one of the things that we saw is that in 2020, when we were measuring our pesticides, so this is imidacloprid here on the left and pyrethroid here on the right, the numbers just kind of fell off the cliff. Using the exact same methods, using everything the same that we did, same people running the assays. And so this was really dramatic. I'm not sure if I quite believe this is so strong, but if it is, then maybe it's possible these bioreactors were maturing over time, and the constant supplementation that we were doing was having a really large effect on them in changing the bacterial communities. And so, you know, we saw a really big effect on pesticide concentrations in this case as compared to when we hadn't added bacteria over here. One of the things that we also noticed was that what was interesting is if we looked at the community of bacteria and looked at what proportion of the community was made up of pesticide remediators, what we saw was that actually the more mature the community was and the more we added remediators in there, we actually noticed them as being a smaller proportion of the community, which might seem odd if we're remediating a lot of things out of it. Where are all the remediators going? What was another interesting thing is if we look at how the community was made up, when we take all our samples and we kind of put them in this two-dimensional space and look at, you know, how many of these different pesticide remediating bacteria of different types of different genuses, you know, what are the explaining this difference? Well, down here, it's a little hard to see, but this is basically where all the bacterial additions are these data points in this area. And this area up here is where more of the data points were from post additions. And we can see that this Burkholdia group is really kind of driving this pattern. And these are the remediators for permethrin. And so we think that it's some of these pyrethroid remediators which are actually driving this pattern, and that we've seen some significant changes, and that's why our pyrethroid concentrations are have now gone down. So it also may be that a lot of the genes that these bacteria have, they've just been sharing with other bacteria in the bioreactor. So we don't might not see those specific bacteria, but their genes may be horizontally transferring to other organisms. We've got a lot of ongoing work. We're going to be looking to wrap up this project, hopefully by the end of June, maybe beginning of July. We're also looking to continue on work on this project. One of my graduate students, Connie Machuca, is going to be looking more closely at some of these community dynamics and how these different types of organisms and how these organisms may be affecting the genomes of these communities and these bacteria, what that process looks like. And so she's going to be working on that this summer as well. We've also been developing some new methodologies for testing for bacteria, for pesticides, trying to find a cheaper, better way to do it. It's a really pain in the butt process for us right now.
Nathan, you've got 5 minutes.
Thanks. And what we have really been able to do, which has been especially useful, we're also trying to take our methods and apply them to some other systems, looking at fluprodiferone just as a way of seeing whether or not what we're doing, we can apply it to other pesticides very easily. Can we look at other targets that people might be interested in? But one of the things I just want to emphasize is that the funding that we've gotten through Leafy Greens has been instrumental in us continuing to get funding to fund all these other projects. So we've been able to use it for match and pull in, more than $300,000 in matching funds from the CSU system. So just a huge help to be able to have funding from Leafy Greens to support the lab. Lots of people to thank. Definitely want to thank the organization for the funding support. The CSU Agricultural Research Institute, where we've got a lot of this match funding. The CDPR for their continued support. And also our Undergraduate Research Opportunity Center. This work has supported, at least like 12, 14 different undergrads, 3 to 4 graduate students working on this project as well. So been a very, had a lot of impact on a lot of people. So that's what I've got for a presentation. I'd be happy to take any questions or comments that people have.
There's definitely some questions too in the Q&A and the chat. Let's do the poll really quickly, and then we'll get to some questions. We'll give everyone a few minutes to log their responses. We'll give them 30 seconds.
And again, if for any reason you can't see the poll, sometimes there's pop-up issues, you can go ahead and post answers in chat if you want.
Would you like me to start answering the questions in the chat in the Q&A, Jennifer?
Yeah, go ahead. You can see both. I know it's hard. The panelists can't actually type in the Q&A. So you've got some questions from panelists in the chat, and I see something in Q&A as well. Yep.
Yeah. So I'll address Suzanne's question first. So she asked, after completing your work, how do you see your findings being applied by growers? I think that they can hopefully be easily applied. If what we're seeing is an effect of a maturing bioreactor becoming really efficient at removing pesticides, these are not very difficult entities to put in place. We built ours above ground, but the goal was eventually to put it into drainage ditches. We just did it above ground because we didn't want to put it in like a permanent structure into someone's field that they might not want in the long term. But we think it could be a really effective tool and a really inexpensive tool, one that won't hopefully take up very much space and really be a nice mitigation tool. The applications of managing microbes right now, we're we're doing all that stuff ourselves. Obviously that would have to be, if it was to be scaled up into a larger scale, would have to figure out a way to do it. There are, culturing, there are ways that we could involve people who could culture on industrial scale if we really had to. But for now, the lab can grow kind of what we need. If people are interested in it, they should just contact me, and we'd be happy to help people design and implement a system themselves. We'd like to try to find some funding too to put in a few more of these so we can replicate some of what we've been doing, especially for these community studies that Connie wants to do this summer. There's some other, how can we, Michael asked about how can we be sure about the pesticide concentration reduced by bacteria rather than binding to the wood chips? Yeah, this is an ongoing issue. So is it just being trapped by the wood chips, right? It definitely could be the case. We've done some experimental work to show that both happens. So it looks like that they are caught, but they're also being remediated. So, I think, eventually we have to look at it. But the nice thing is that if the bacteria are in there, we would expect that the bacteria would also be breaking down the wood chip, the pesticides in the wood chips. It actually works okay if the pesticides bind the wood chip, because if the bacteria stay in there and are able to remediate it, we would expect that they'd be able to break it down over time too, which would be a nice effect. David asked about the effective age of the bioreactor, and I think that's definitely a case. We could be looking at an instance where we're seeing a succession and maturity in this community. Bacterial genes spreading throughout the community, remediating genes spreading to lots of other organisms, and that actually having a big effect on bioreactor becoming more efficient.
Nathan, there's one more question that just came to me from Krishna. When were the bacteria added to the bioreactor, and how soon after were the pesticide levels measured in the water?
Yeah, so basically we, when the first time we did the experiment, we added them in, and I think the next day was the day we started sampling, and that's pretty much the pattern. We added in on a Sunday, and then we'll go in and we'll start sampling on a Monday. That's in best case. We're kind of dependent upon when the pump system runs. So sometimes they sit a little bit longer, sometimes they don't. We were always worried about them being sort of flushed out faster than we could do it, but we just tried to stay consistent with it. So we always add on the same day of the week, and then we sample when the water runs.
And then Nathan, we'll wrap it up real quick with the poll. If you want to just address, it looks like everybody did pretty well.
Yeah, they were both true. So imidacloprid is definitely one of the ones we target. It's probably one of the easiest ones for us to measure too. It's water soluble, so we would expect it to move through the system fairly easily, unlike the pyrethroids, which might bind to things like the wood chips a lot more. As for the bacteria, yeah, they're all naturally occurring bacteria. So these are all things that have picked up these. We've isolated them from the environment, and they just have naturally picked up this ability.
Great, thanks. And we'll there's one more question in chat, and Nathan, I'll let you answer. Will you just type in an answer, and then that way we can keep moving on the agenda?
Definitely.
And yeah, and I'll say it again for everyone. If questions come up, as we're moving along, just keep typing them into the Q&A, and we will keep having presenters answer those, and we can circle back at the end too. And so with that, Nathan, if you'll stop sharing your screen, and then we'll get David Still's presentation up. David, good morning.
Good morning, everyone. Let's see, let me share my screen with you all. Okay, do you see my slides?
Yes, it looks perfect.
Okay, very good. I'll get started then. Okay, so today what we'll talk about is nitrogen and water use efficiency in breeding. And I wasn't able to be on the program yesterday, so Jennifer, thank you for accommodating me and moving me to today. So I wanted to start with an overview and just kind of set the frame of how we got here and where we're headed. But if you're going to try to improve water use and nitrogen use efficiency, there's three large components to it. It could be at the whole plant level, the physiological components to it, and also the genetic, which is what we're mostly interested in here for our purposes. At the whole plant level, One of the things that'll affect it is the leaf, like the exposure, if it's a head versus a leaf lettuce, stomata, the wax, the cuticular wax, the thickness of the leaf, the density of the leaf, color even. And of course, the root length and biomass are a couple of easily imagined components that would affect it. At the physiological level, we know that photosynthesis and nitrogen metabolism are the two main components that are going to be affected, but not the only ones. And the interesting thing about them is that they're interconnected. So why a plant needs nitrogen is to support photosynthesis. So somewhere around 70%, and in some plants it's even higher, 70% of all of the nitrogen that's used in a plant is to support photosynthesis. Literally the photosynthetic machinery, the photosystem 1 and 2. And the genetic components, we always have to contend with the environmental components in that interaction. So the soil properties will affect both water use and nitrogen use efficiency, and the microbial communities that are controlling the nitrification and other aspects of the nitrogen cycle. We know that it's really difficult to get the water and the nitrogen applied evenly on the field, so that really increases the environmental component to it. Now, plasticity is something that all plants have a degree of, and we might mistake that for some sort of a genetic component. We might also mistake it for an environmental component. It truly is a combination of both. So the plasticity in our context here is when a good example is roots. So the soil density will affect how the roots grow. How much water is available will affect how the roots grow and how much nitrogen. So if you start withholding water and nitrogen, one of the things that seems to be conserved across the plant kingdom is that the roots actually get a little bit longer. So that's a plasticity component that also is genetically controlled, and not all genotypes have it. And of course, the leaves have a lot of plasticity in them as well, but today we're mostly interested in the roots. So to put things into the next framework of nitrogen, why do we need nitrogen? What we have here is a diagram showing some lettuce plants that are going to gain weight, so above ground biomass on the y-axis, and the weeks after planting on the x-axis. What we've done is we have 4 different nitrogen treatments that we've labeled N0 through N100. And these are equivalent to what we've done out there in the field of 225 pounds of nitrogen per acre, which is what we might do down in the low desert and some of the lighter sands, down to adding no additional nitrogen. So the plants are just growing on the residual nitrogen in the soil and the nitrogen that might be in any of the irrigation water. And it's really exactly what you would expect, is that you add more nitrogen, you get more biomass. But there's a limit to that, and you can't just continually add that on, and especially in the environment that we're in, with wanting to mitigate the environmental impact of growing our crops. Now, we just wanted to demonstrate something that we all know inherently here, but at the same time, develop an experimental situation here that would allow us to manipulate why we need this nitrogen, and that's relationship between the leaf nitrogen concentration and the photosynthetic efficiency. So the next set of slides are going to address that issue. If we have on this y-axis the carbon uptake, CO2 uptake, which is carbon assimilation, or sometimes called photosynthesis, and then the leaf nitrogen concentration on the x-axis, and then we have evaluated about 10 different genotypes here that we've characterized as either being high leaf nitrogen accumulators or low leaf nitrogen accumulators. So the high nitrogen accumulators have the open symbols, and the low nitrogen accumulators have these closed symbols. And again, this is the leaf nitrogen concentration. And we were using the leaf nitrogen concentration as a metric of the uptake and assimilation of nitrogen from the roots and assimilation into organic compounds into the leaf that would support photosynthesis. And what we said earlier is that about 70% of the nitrogen in a plant is used to support photosynthesis. And we have an R value here of a 0.65. So that shows you with this regression line pretty close to what the theoretical is. The other thing to notice is that we have a lot of variation in the ability in carbon assimilation, and we have a lot of variation in our leaf concentration. So in our next slide, if we reduce the amount of nitrogen by half, the slope goes down a little bit, and we start seeing some spread among the different genotypes here. If we lower the nitrogen by another half, you can see that the leaf nitrogen concentration now starts really getting narrow. And we still have a lot of variation though in the CO2 uptake and assimilation. And this is exactly what we want to find, is those, as we start reducing the nitrogen that we apply to the field and that the plants are able to put into their leaves, that we can still maintain these high assimilation rates. So this, we could interpret this as that these membranes are maintaining their integrity. The membranes of the photosystem in the chloroplast is what's really occurring here. So if we decrease it and add no nitrogen, you can see that relationship here between leaf nitrogen and CO2 is disappeared. But the other thing to notice here is we still have this range, a pretty narrow range in leaf nitrogen concentration, and a very clear demarcation between the low leaf nitrogen accumulators and then the high. So this is that's something that's been genetically conserved in this population. And it helps establish the principle that if you go ahead and select for high leaf nitrogen concentration, you will be able to improve nitrogen use efficiency through carbon assimilation. So you can directly select for carbon dioxide uptake or carbon assimilation. So the next step that we wanted to look at over here was, can we map this? And so we took this approach where we would just use a biparental population, the diplomat and margarita, recombinant inbred line population. And we would feed it water and nitrogen at 225 pounds of nitrogen per acre, which is fairly standard down in the Yuma area. And then half that rate of water and half that rate of nitrogen, and then do the comparisons. So the metrics that we would look to measure water use and nitrogen use efficiency were, for nitrogen metabolism, the nitrogen isotope discrimination. And for the integration of water stress over the growing season, carbon-13 discrimination. And then we looked just at the leaf and soil nitrogen concentration. And again, looking at that leaf nitrogen concentration as an integrator of uptake and assimilation, and in other words, nitrogen use efficiency. Some of the data that we'll show you here for the mapping is off of 3 production years down in Pomona and two actually in Yuma. We'll show you some data from field and controlled experiments. And then simply because we shouldn't draw all of our conclusions from biparental populations with a very limited genetic base, we screened a bunch of germplasm, selected the best types, and made 7 different interspecific F2 populations. And that work continues, where we're making selections and backcrossing. In terms of environmental variation, what you're having to deal with, we're showing you the nitrogen metabolism on the y-axis. And this is more or less, it's nitrogen isotope discrimination, but just suffice it to say that we can discriminate using high numbers versus low numbers. What we've done is we've ranked in one of the 3 years, we've just ranked the values of nitrogen metabolism from high down to low. And then for the 2nd and 3rd year, we just map that on top of there to look at what that pattern is. And clearly we can see there is no pattern. So this is that environmental variation that these plants, when they're out there in the field, they can alter their metabolism due to whatever environment that their roots are in. And then if we do a more controlled experiment, we can get a little clearer picture of what's going on here. We've done sort of the same thing. We grew the population at N100, meaning that nitrogen is not limiting their growth, and then half that amount. And we ranked the nitrogen metabolism values from high down to low. And then we looked at those same, where we retained that ranking in that order. You can see that there's actually no relationship between the metabolism that these plants use under high nitrogen versus low nitrogen. This means that if you want to improve nitrogen use efficiency, you need to make all selections and do your whole experimental setup under conditions of low nitrogen. You can't make any selections the way we do it normally under the current growing conditions and try to make any progress in improving nitrogen use efficiency when you start to limit nitrogen. I want to be clear here, though, that if you give these lettuce plants all of the water and all of the nitrogen they possibly want, they do great. We already know that. But once you start limiting them, then we have to change the genetics. Now, we were able to map these. This is just a schematic showing the circular chromosomes in these outer ring is the different linkage groups that Richard's group has we borrowed this map from them, and then we just mapped onto them the QTL that have been associated with different components of nitrogen use efficiency and water use efficiency. And what we find is that there's a lot of QTL that are we identified. There's a few with fairly large effect, and they cluster in linkage group 4 and linkage group 5. So these are components that have to do with head weight. They have to do with both water and nitrogen use efficiency. And then the same sort of thing over here in linkage group 5. Now over here, there's some others that are solely due to water use efficiency in linkage group 7 and linkage group 9. And the take home to this, though, is sure, we could map them, but the environmental component is large. We didn't have any of these that showed up year after year in the same spots. So again, focusing on what we want to do is look at and see what kind of variation there is, make sure we're capturing all the variation and the greater germplasm. We made the interspecific crosses using different Seriolas, Saligna, and Verosas as part of the parents, and then crossing some against the some high leaf nitrogen concentration genotypes, and then developing these F2 populations. And again, selecting for nitrogen use efficiency defined as high leaf nitrogen concentration genotypes. If we just look at the population means of those in the FF2 populations, and we compare that with roughly about 100 commercial cultivars that were growing at the same time, then we find out that at this population right here, these are about 22% higher than the best commercial cultivars. And we have maintained that in F3s and F4s as well. And now we're in the F5s. So, and starting some of the back crossing. So that high leaf nitrogen concentration was highly heritable, and we again, we'll have some, we're making back crosses to some of the cultivars, and we will make those selections available later in this year. Now, this is a slide to kind of remind us all that we already kind of know. This is just a field where we were growing the lettuce, and we put our tensiometers at 6, 12, 18, and 24. And the water potential here on the y-axis, this is where the water is, and it's moist down here and drier here. And as we go through these different irrigation cycles, clearly we all know that at the 6 and 12 inches, the water is evaporating out and being used by the plants. So that's evapotranspiration. But down here, the 18 to 24 inches is where all of the water and the nutrients are. And boy, wouldn't it be great if all of our roots were down there. But no, most of our roots are actually up here. But we also have these high nitrogen leaf accumulators, and we had to look at the roots because that's the next step of this whole thing is, again, one of the main components we think that should be addressed. So we looked at a high nitrogen accumulator, and this thing has roots that are about 28 inches long, and when it's grown under N100. But when we grew it under N50, and it still accumulated nitrogen in the leaves quite well, it lost a lot of its biomass and length. But when we compared that against one of these low nitrogen accumulators, and this is the general trend, they don't do well under high nitrogen, but under low nitrogen, they really improve their biomass. So
David, you've
this
got
is
5
the step
minutes.
we're going. Thank you.
Sorry, David, you've 5 minutes.
Perfect. Thank you. So just to kind of show here, to conclude everything and sum it all up, we did find a lot of QTL that are associated with water use and nitrogen use efficiency, but only a handful have major effects. We know that the environmental variation in nitrogen use efficiency is tremendous. Again, it could be an uneven application of nitrogen, but also the soil microbial communities. And this is where Nathan would come in. High nitrogen accumulators actually have higher rates of carbon assimilation, so we can select on that. There is an allelic basis that looks to be highly conserved. We know that the non-domesticated species are good size sources for nitrogen use efficiency. We just don't know why. But we are going to backcross this, that black box. We'll backcross and release those high nitrogen accumulators later on in the year. And what we want to find out is, can the root biomass be combined with the high leaf nitrogen concentration? Right now, they seem to be separated. We have tried to use this ground penetrating radar. We showed you some data last year on that. It didn't work out well for finding biomass. It's maybe it could be used for the root length, but right now we're not super hopeful that we'll have a high throughput method, and so that means we have to turn to the shovel, unfortunately. I got to thank the folks in the lab that did the work, Dr. Yu, Dr. Stroud, master's student Justin Medina, and of course down there at the UMAX Center, Jesus Pereira is really a lot of help down there. Funding from the California Leafy Greens, the CDFA Specialty Crops, especially the USDA Specialty Crops, and the CSU Agricultural Research Institute. And with that, I will stop my screen sharing and answer any questions if anybody has any.
Thanks, David. I don't see any questions at this time, but we'll just keep monitoring the Q&A and the chat. Yeah, I don't see any chat at this time. So thank you. And you don't have a poll question because we can get DPR credits for your work, but I'm sure that's part of the CCA credits. So okay. I see Steve Klosterman is ready to go. And okay, we see your presentation, Steve. It looks great.
Okay. All right, thank you. So today I'm going to be discussing what we've been doing with spinach and lettuce downy mildew detection and epidemiology. I'm having trouble advancing the slide.
Sometimes do you have 2 screens or 1 screen?
I have 1 screen.
Oh,
if you go down to the bottom left corner, yeah. Is there arrows anywhere? Can you click there? There.
Yes.
And then once you click that once, it tends to wake up, and then you should be able to use the arrows.
All right, thanks a lot. I've never seen that before. All right, so our objectives are these. So I'll touch upon the first 2, the third one here, and the completing of the biopesticide testing. As some of you are aware, in 2020, we didn't have natural disease, so it's hard to test that. And the nitrogen dioxide seed treatment, as you know, that some of you know that has been pretty effective for killing Verticillium dahliae. But we are still working with a representative of National Organic Program to try to get that approved. So we're working through the regulations. And the last one is what I will begin with today, is determining the inoculum load of downy mildews in the Salinas and Coachella valleys. And just to show this again, I think maybe Jim had some of this yesterday, but the symptoms of downy mildew are very striking, shown here, these yellow lesions on the top of the leaves, and underneath is what we call in plant pathology the signs. So you can actually see the pathogen. Those are signs. So the spores, there can be up to like 10,000 of these per leaf. So these are released airborne into the environment, and then we're able to detect these, and the spores from Bremia lactucae, the lettuce downy mildew pathogen, are about the same size, about 30 microns. So we're able to detect these using these impaction spore trap samplers. The impaction spore trap sampler has a rotating arm at the top, and this arm contains two of these steel grease-coated rods. So these are coated with a very viscous grease, and the spores will actually impact when this arm is spinning. And we couple this with a quantitative PCR multiplex assay. So now what we're able to do, we completed this in 2020, is simultaneously detect Peronospora fusa and Bremia lactucae using the mitochondrial markers that we had previously developed with Frank Martin and Alan Van Dynes. So looking at lettuce downy mildew, as you can see here, this is on the left-hand side. Here's the number of spores in this non-linear kind of scale over February, March, and half of April. And so in both the Salinas and Coachella valleys again, so this is where we were examining them. As you can see, there's maybe an uptick in both Salinas and Coachella valleys through February through April, as we might expect because of the increase in planting, of course, in Salinas, and also these crops are maintained through April in Coachella. But what is really striking here is what's going on in Coachella Valley relative to the Salinas Valley at a similar point in time in October and November. Down here at the bottom of this graph on the right, you see that these are 0 values. So essentially, not having the crop in October and also potentially these very high temperatures. The average high temperature in Coachella Valley at that time was 97 degrees for that month. And relative to the Salinas Valley then, there is actually at the same time very high levels of the lettuce downy mildew pathogen spores that are floating around. And so one could argue then that that it's the sheer volume of the crops in the field that's actually driving these numbers of the spores that we see. And for spinach downy mildew, it's a similar kind of trend. So in October and November, it looks like it's being cleaned out of the Coachella valleys, the Coachella Valley rather, and in Salinas, we do see, as we've demonstrated before, actually, that February through March, we have pretty high levels even before the planting. And as we go to April, at Coachella Valley, there is this little bit of a trend upward in the spinach downy mildew. So we are currently deploying the spore traps in Salinas, Imperial, and Coachella valleys. This is with collaborators Alex Putman and Ali Montazar in the Imperial Valley. And so all the spore trapping that we've done to date, we've used these impaction spore traps, as I showed earlier. So what I wanted to also show you is this new type of spore trap that's being produced by Root Applied Sciences in Berkeley, California. So this is called a cyclone spore trap. And so using the cyclone spore trap, the sampling is much easier. And so making this the spore trapping one step closer to potentially automation. Using this lure lock type system at the bottom of this bottle looking structure here, the air is taken in through this instrument and funneled into a cyclone, and the small particles like the size of the spores will drop out into this tube. And so it's very simple to collect these versus taking off these stainless steel rods, replacing them with and putting grease on them and doing that each time. And so what we did was use the two types of spore traps to compare. And we took a trap of each type and placed it on the north and the south edges of a sporulating field plot. This is Charlie Brummer's field plot that he mentioned yesterday from near Salinas in November, early December. And so the technician and I were stunned by our results. So these new types of instruments appear at this time to be about 10-fold more sensitive. You might ask, why is this? So the company estimates that their volume of air sampled is about 1,000 liters per minute, whereas these other older spore traps that we've used is about 50 liters per minute. So I want to emphasize the advantages over the impaction spore traps that we've been using. So we need to repeat this, of course, but it does appear to be more sensitive in the initial trial. And it has this ease of sampling that makes it so much nicer. And I've always said that the growers or PCAs, that it would be very difficult to use those stainless steel rods and replace those and keep everything clean and so on. This makes that much easier. It is cheaper to deploy over the long term. These newer cyclone traps are about $3,000 per unit, and these older traps are about $400 per unit. But when you consider in the grease, the sampling rods, and some of the wiring problems that we've had over the years with the older traps, it would be nice if we're able to use these routinely, the newer traps. So they are working toward commercialization of these types of traps. I think that they have some deployed in some vineyards currently, and they're working toward automation so that it could actually be the signal from this tube eventually to be transmitted wirelessly to convey the information on spore load. So what is the value? It's always good to wrap this up with the value, the applied value. So we're trying to refine the disease forecasting to improve that. If we can demonstrate, how much at what time, under what conditions, then that can be useful in itself to the growers. And potentially, and I emphasize potentially, in down the road, genetic markers could be used to identify characteristics in airborne Paranospora effusa populations, sets of races, for example, or fungicide resistance. And that's where, of course, that what Richard Mitchellmore brought up yesterday in association with Alan Van Dynes' group that's leading it, developing those the high quality sequence of Paranospora effusa sequences and being able to fish out some of these characteristics is key. It's a very important component of the work. And I'm very happy that Jim Carell and I with Kelly Clark, the postdoc working with us, is also contributing to that. She's been doing some race typing. So the cost effectiveness of the spray programs rather can be improved through disease forecasting as well as fungicide resistance management if we can target the fungicides better. So switching gears to the oospores. So shown here at the upper left, these are oospores of Peronospora effusa. These are the long-term survival structures of the organism. And they are indicators of sexual reproduction. So there's two mating types in this pathogen. And we have been able to observe these oospores routinely in the leaves of some cultivars in spinach in California. And as Jim mentioned yesterday, Jim Carell, recently several cultivars down in Arizona too in that field trial. So they are present in about 17% of seed lots, and those from seeds and leaves germinate. There's a germinating oospore shown up in the upper right of your screen. And as I mentioned, they are long-term survival structures. So this is why we are concerned about their transport worldwide and how they might be the disease then might be transmitted on the seed. And so we've been investigating the seed transmission. Shyam Kandal, who was a postdoc previously with me in Bejuan Mo, and Kelly Clark, who's now taken this over, postdoc working with me, and Jim Carell. So what Kelly is doing is using these plant isolators to plant the infested seed, oospore infested spinach seed. And these plant isolators, of course, they're valuable for this project because they lock out the external airborne spores that I mentioned earlier that could contaminate the whole experiment. So if it's locked in there, then we know that the source of the downy mildew must have been from the seed. And so in 2019 and 20, I mentioned this before, earlier at the mid-year meeting and the last meeting in March. So Shyam Kandal was able to identify downy mildew on spinach growing in these isolators in 2 separate experiments from infested leaves. But the negative control was also came up as downy mildew positive. The negative control in this case was viroflay, which is susceptible to all the different races of downy mildew. But at the same time, that those negative control seeds were also PCR positive. So it's potentially we missed some oospores that were hanging out in those seeds as well at a very low level. So essentially I just ran through that at the top here, but in the winter of 2019 and 20, we did not observe downy mildew from the planting of 2 replicates of 2 oospore infested seed lots. And a seed lot that Jim Carell had found where there was downy mildew that was reported in a field. So why do we think that might be? So there is potentially declining viability of the oospores in a couple of those seed lots that are now about 3 or 4 years old that Shyam was using earlier. And we have to keep in mind, of course, that this is only about 0.04% of an acre that we plant each of these in the isolators, these lots, these samples of these lots. So it's maybe a numbers game, not high enough numbers. As we know, the distribution of the Peronospora effusa in in the seed lot is not necessarily uniform from some of the data that we have.
Steve, you have about 5 minutes.
Okay, thank you. The final objective that I wanted to discuss was, does spinach downy mildew cross-infect the weedy plant species that's shown here, nettleleaf goosefoot? Shyam Kendall had brought this to my attention, and I thought it was a good thing to test because there was lots of downy mildew on this nettleleaf goosefoot, which looked the same as the downy mildew that you would see on the spinach, and they were growing side by side. So he did these experiments where he inoculated the nettleleaf goosefoot pathogen on spinach and the spinach pathogen on nettleleaf goosefoot. And the answer is no. There's no disease on nettleleaf goosefoot. So it's important to check these out every once in a while, these weeds, to see which ones might be acting as a reservoir for any of these pathogens, including spinach downy mildew. But this is supported by lots of research that goes back 50 years or so that spinach downy mildew only infects spinach, host specific. So in conclusion, we've developed a multiplex assay in 2020 for simultaneous detection of lettuce and spinach downy mildew. The airborne lettuce, spinach downy mildew pathogens seem to be always present in Salinas Valley, backing up some of what we've done before. In the winter months, this is potentially related to the production of freezer spinach, home gardens. In the case of lettuce, well, maybe wild lettuce contributes to that. There's the lack of crops and high temperatures in the Coachella Valley apparently just cleared out both of these pathogens in October and November. And the newer cyclone traps that I showed are 10 times more sensitive, at least in that initial trial. And so we are ordering 6 of these units for comparisons, and hopefully we will start to use those really soon. The oil spores of spinach downy mildew are present in about 17% of spinach seed lots. So as I mentioned, these are the survival structures. So this explains possibly why the disease occurs in new areas. And spinach downy mildew disease transmission tests were not reproducible this time. But I wanted to make that note that it would be unusual actually if this Paranospora species was not seed transmitted, as so many Paranosporas are. I think some of you are familiar with even a published report from spinach indicating that it's seed transmitted, but the numbers are so low. The percentage less than 1%, sometimes 0.1%. So in these in that spinach study, so it might be just the numbers game that we're playing. And the spinach downy mildew pathogen did not infect weed species tested. So it looks like it only infects spinach. So for the acknowledgements, current team members are listed at the top, collaborators, recent collaborators, and of course the funding is, thank you, California Leafy Greens Research Program.
All right, great. Thanks, Steve. Let's get the poll questions up and we'll give people some time to answer the poll. And just to keep us on track, Steve, you do have 3 questions so far in the Q&A, and maybe if you will just answer those in that Q&A section, just maybe type your answer. I think you might have answered the first question in your conclusion, but feel free to expand.
Okay.
Thank you. And then we'll just
explain. Okay, you just want to go on?
Yeah, I think we should just do, let's just try to keep on track. We did a great job yesterday, and I'd like to repeat that.
Yeah, we're on time, I think, right now.
Yeah. We'll give a few more seconds here for this poll.
About 20 more seconds.
Okay.
We've got 47% voted and they're still coming in, but 10 more seconds.
And just
Okay.
just a reminder for all the PCAs, these are the questions you will see for your continuing education credits.
Okay, very good. 72% got the first one right. 17% of the seed lots contain these oospores. Yes, does not infect weedy plants. So true, 75%.
Great.
Good.
Thank you.
Okay. So
Yep.
I'll stop.
Okay, great. And with that, we've got JP Dendor Arias is next. And I believe JP has some students on as well. So welcome CSUMB students. Great. And good morning, JP.
Good morning, everyone. Can you hear me well?
Yes.
Awesome. Thanks everyone for being here, and Jennifer and the board, thanks for the invitation. I'm going to present some of the work that we've been doing over the last year, investigating the biology and management of Pythian wilt of lettuce. This is work that I do in collaboration with Richard Smith, which without him, we wouldn't have been able to do any of this work. Can you see my arrow on the screen?
No.
No? Okay.
We seem to have pretty good luck with that little laser pointer if you have that option.
Yeah. Let me.
Oh, wait, I can see your little tiny arrow. I do see it. It's small.
Okay. Okay, I'll try to point out what I'm showing. Okay, so Pythian wilt is a disease that everybody probably in the audience have heard about from over the last year. This project was proposed in response to an increase of disease occurrence that we have been seeing over the last few years, but we have never seen up until last year seen anything like to the magnitude of damage that we encountered last year. There were fields and growers reporting up to 100% no harvest yield losses in many cases. So what we proposed to do was to characterize the local Pythian populations affected lettuce and explore what were some of the factors that influenced the geographical distribution and establishment and development of the disease here in the Salinas Valley. So we have three main objectives that, and I'm going to walk you through each of them. The first being monitoring the occurrence of Pythian wilt of lettuce here in the valley. So Pythian started last year rather slowly, and we found it the first time around April, at the end of April of last year. And it was just like mainly like like a very occasional situation, like some fields here and there. And the first field that we found was this one that was visited by Richard Smith, and he noticed that some of the plants were lagging behind. They were stunted, and they were, when he pulled the plants, he noticed that there were some rotting. So he brought the samples to my lab, and we found that actually these two plants that you can see here in the middle were actually infected at a very, very late stage, actually, even though the above ground portion of the plant was not really that much smaller than the plant that we did not get the pathogen from. We also collected some other plants from the same field that were from different varieties, and we were not able to find the pathogen there. So at the beginning, it was just these two samples that we got. But eventually the outbreak is spread all the way from King City to Castroville. So we started the sampling. So Reacher would be our main person who would go to the field, collect the samples, or grower or PCAs also would provide some of the samples to Reacher. He would bring it to us. We would watch the roots, take care of them, and then play them on following previously developed methods using selective media. And what we found, we end up actually collecting samples from 55 different locations across the valley. We got over 300 plants that we processed last year, and we ended up plating over 2,000 individual root tissues or sections, including both tap and lateral roots that were plated and analyzed individually. So what we found in terms of some of the conditions and practices that were associated with more with the disease was that like the large majority of samples came from these shoulder long soils, even though you could see that in the other soils that where we got samples, any kind of clay loam or sandy loam were also it seems like the disease was also showing up in those fields. And that I want to point out that these are just samples that we got. It doesn't mean that the disease, so maybe some of you have were able to see more disease in some of these in your own fields that have this particular type of soils. But what this is just the frequency distribution of how many of the samples, the total samples that we got, how many, like where they came from. And the majority in our case was sugar loam. In the same case, we got the majority of samples that we received and collected were from conventional production farms. And even though we got also a few from organic fields, but again, that doesn't mean that the disease occurred or there was something in the conventional production that necessarily was more conducive. We did not test that question. It's just a matter of the frequency of samples that we got. Then we started also conducting more field visits and field evaluations, and we were noticing that in the majority of cases, the tail ends of the fields were often more severely affected. And that makes total sense if you think about that this is an oomycete pathogen, or also known as water molds. So they relied and need or depend on our favorite, for with saturated soil conditions favor the pathogen and the establishment of the disease. So often the end of the fields in both fields with drip and also with sprinkler irrigations, we noticed that in many of the cases, just end of the fields were more heavily attacked. However, when we look at sprinkle versus drip irrigation, we didn't really see main difference. We tended to see more sprinkler, more samples coming from fields with a sprinkler irrigation in the spring, so in the first crop of last year, but by the fall, we were equally seeing the disease coming from both fields with a sprinkler and drip irrigation. Here, it's also what we observed was that older plants in general tended to collapse later or faster in the crop cycle. So this is consistent with some of the work that has been previously reported in observations from Steve Koike and some of others that have reported that pythian will tend to attack plants at a later stage. However, in many cases, we also start seeing, especially in the fall, we started seeing plants at different stages being attacked. So you can see in this graph that all the samples that we got from April to July were just very close to be harvested, plants that were fully developed and were taken down just at the end of the season. But by the fall, we got a little more distribution in getting samples from plants that were at later stages, but even at some of the more younger stages as well. Field observations also allowed us to see that there was a varietal resistance that we noticed. There were particularly multi-leaf plantings. There were certain varieties that show specific, higher susceptibility to Pythian. But we also, so that was mostly in like green leaves, and there were like, there's a long table that Richard Smith put together and is posted in one of the blogs at the UCCE website that has the different ratings that we got for each of those cultivars. But then again, in the fall, we started seeing more, even some differences in head lettuce varieties that show different levels of susceptibility. And the only one where we did not really see a main difference were in romaine. Romaine were attacked by Pythian. We got a lot of samples from of romaine, but we were not able to notice any particular variety that was more or less susceptible. But in general, something that was consistent across the season was that red leaf types were generally more resistant than especially the green leaf types. However, if you remember from my first slide, I told you that our first sample was a red type. So that doesn't mean that all red varieties were resistant. As a matter of fact, you can see here that in this picture, there were some red plants that were infected, but in general, they tended to be a little more less susceptible, or not hyper susceptible like some of the other varieties, like you could see in this picture here. So the second objective that we had was to look at determine the sensitivity of Pythian to some of the commercial products and the efficacy of these products against Pythian wilt. This is in particular, we've been doing some in vitro sensitivity assays to see whether the local populations of Pythian are express any there any difference in the sensitivity to mefenoxam, which is the active ingredient of the redomil, which is one of the main products used for the control of oomycetes. So this is work being done by Cassandra Tice, who's a student in my lab. And what she does is using media that has been amended with redomil at different concentrations, like non-amended, and then at 110 and 100 micrograms per milliliter. Then we add the corresponding isolates. We screen the sensitivity of the isolates to the different concentrations of the product. So she puts an agar plug that she takes from these cultures, put it in this amended media, and then she tracks the growth of the cultures for over time, and then she measures the growth of these cultures in each plate over time, again at days 2, 3, 5, and 7 days after the initial cultivation of the isolate. So then with that information, she's able to, by the end, to come up with some assessment or classification whether the isolates were sensitive, meaning that the growth at the highest concentration was significantly less than in the unamended control, or whether they were insensitive if they actually were able to grow even in the place amended at a higher concentration. So what we have found is that in general, she's only been able to do pretty much a third of all the isolates that we have in our collection, probably a little bit less than a third. But so 35 isolates, what she has seen is that the growth of the majority of these isolates is reduced by the presence of this product in the media, particularly redomil gold, at both the 10 and 100 micrograms per ml of active ingredient dose. However, what we also noticed is that there was insensitivity to this active ingredient was found among field isolates of Pythium that cause Pythium wilt. So this is something that we need to keep an eye on, and I'm not claiming that these isolates are resistant, because we don't know if they were previously sensitive and they became resistant. So that's why we are calling them insensitive. But what is important to think about is that keep in mind that we have to be strategic and not overuse this chemistry, so we do not push or increase the selection for these insensitive populations, and then start building their populations in the soil. And also consistent with some of the work that has been done before, you can see here in this table that the average growth of the organism on these plates, you can see it here as the growth, how much of the growth was inhibited compared to the control at day 2, 5, and 7. And you can see that in both concentration at 10 and 100, the sensitivity or the inhibition of growth start declining over time, which is again consistent with some of the work that others have shown, that these products don't, particularly this chemistry doesn't stay very stable, and it tends to be degraded over time, even in vitro conditions. But more importantly, even in the soil, methylaxyl or mefenoxam tends to degrade it faster due to different physical, chemical, and biological factors, including biological activities similar to what the work that Nate showed earlier.
JP, you have 5 minutes.
Yeah, thank you. So we conducted fungicide trials, and then we look at whether Pythium was, Ridomil was effective against Pythium. What we were able to see is that in two of the trials, the applications at Rosette and Thenin were actually contribute to reduce the incidence of the disease, which is exciting. We're going to continue this work because currently these are times where that are not included in the label of Ridomil, but we are testing whether increasing these evaluations in the field this year. And then that takes me to the last objective, which were characterizing the isolates that we have collected. We have a collection of over 150 cultures, pure isolates collected from last and this year from coastal California and Yuma. And I want to thank Steve Koike for providing some of those isolates, as well as Stephanie Zielinski from Yuma. What we have seen is that we've done all the isolate morphological characterization of those isolates, looking them under the microscope and looking for the signature structures. We have found that the majority of them coincide with the categories previously described for the Pythium cineratum, which is the species of Pythium previously described by Steve Koike and Mike Davis in California, associated with Pythium wilt. And that's also consistent with a small survey that the CDFA conducted that reported that the majority of isolates that they got were Pythium cineratum. However, we got some isolates that didn't match that classification. And we have also noticed some variability in the growing patterns. So we are looking at them using molecular characterization, but this is work that we're still doing. And then I just want to summarize this quickly. As you can see, this is a graph that we got from the Salinas South station. You can see here air temperature and soil temperature. And what you can see is that at the beginning of May was when we found the first or the earliest infection were detected. And then by later in the summer, we start seeing the infection keep increasing, or was still moderate, but not as bad as it got later. But you can see that there was a trend of increasing soil temperatures along this way. Then we got two heat events that consisted of over 2 or 3 days of temperatures over 100 degrees. And you can see that that was really the beginning of the widespread severe infection that we started seeing. So what I want to point out from this graph is that these temperatures between like high 50s and high 60s were really the temperatures that really are conducive for the occurrence of this disease. And then the air temperatures about 85 degrees are the ones that are contributing to see the decay or the melting of the plants in the field so drastically. So just quickly, I want to walk you. This is a proposed life cycle of Pythian well. What you can remember is that spores are the surviving overwintering structures that are resistant to desiccation and can survive even in the absence of the crop. These spores germinate when the conditions are right, germinate, and then they can either infect the plant directly or produce spores that can travel through water, particularly in irrigation and rainwater. The infection occurs through the root tips and wounds. The disease development is favored by temperatures, again, that are around or above 60 degrees in high soil temperature. We also, what you would observe in the field is a reduced root density and shorter plants, shorter roots, and in general rotting of the external parts of the roots, as well as older leaves would wilt and have smaller growth, and there would be reduction of yield. The disease is often prevalent in the wetter patterns parts of the field, and then even though you see that the plants are dead and nothing is happening there, those are the favorite spots for the pathogen to survive. So they can remain in debris, in dead roots, as well as in the soil. So it's important to keep that in mind if you are leaving your fields, your plants in the field. Then we have a series of new projects. Some of them were mentioned yesterday by Richard McMore and Kelly Richardson, but we're also doing some work with Jim Correll, looking at the pathogenicity and host specificity of Pythian isolates from spinach and lettuce, and also starting a project with Richard Smith and Michael Kahn, looking at the effect of irrigation on management of Pythian well. And with that, I would like to thank all the collaborators and people in my lab who have done all the job, and of course the board for the funding. Sorry, Jennifer, if I ran
that's great.
over.
No, thanks, JP. And we'll go ahead, and I think JP has a poll that we need to put up, and I know, JP, there is at least one question, maybe two questions now for you. So if you could just answer those in the Q&A section, that'd be great. And just as a reminder for everyone too, when those questions go away, maybe everyone's figured this out, but there's a little answered tab you can click on to then read those answers. And thank you, JP. This is great. I think that last slide you showed with collaboration is awesome. We've got a lot of great collaborations happening, and we're super happy that CSUMB has you and Elizabeth Mosqueda now. That's great. That's great. Let's see. So Kelly, did you stop the poll? I think so. Yeah. So there's our poll results, JP, and it looks like 100% on that first question. That's great.
Everybody got it right. The spores are the structures that allow the pathogen to survive in the field. And then for question number 2, all of the above were correct. Yes, the disease is affect different types of lettuce, or romaine, leaf, and head. Also, there are different patterns that we could see in the field, but especially in the wetter parts of the field, the disease tends to aggregate. And then the symptoms include all these different characteristics: stunting of plants and rotting of the exterior layers of the roots.
Great, thank you. So we'll put that away, and Bill Wintermantle is our next speaker. And Bill, no pressure, but you're standing between us and a break.
I'm used to going right before lunch, so this is no different than
It's
that.
not the same, but kind of close.
It is close. So yeah, I don't think it'll take, might not even take the full time, but I'm going to give you a little
Great.
bit.
All right. So
see.
you want to share your screen?
Hang on a minute. Let me pop up the talk. Let me make sure we can do this. Okay. All right. Can you see that?
Yes. It's not in presentation mode yet.
It is now.
There we go. That's
Right.
perfect. Thanks.
So most of you are familiar with lettuce dieback. It's obviously a disease that's been around for a very long time. Just to put it in perspective, it probably dates back to the 1930s or earlier under a different name known as brown blight. We have done a number of studies through the genetics program that suggest that those two viruses are the same. So this disease has been around a very, very long time. A source of resistance to the disease was developed by Ivan Jagger back in the late 1930s that became incorporated into iceberg lettuce, but not other types. And then when the planting of large amounts of romaine and leaf began in the 80s and 90s, the disease kind of reemerged, and that's what we're seeing now under the different name, in this case, lettuce dieback. Most of you are familiar with what the disease looks like. As I said, this is an example of a typical field. You'll see plants of varying stages of growth with necrosis. Unlike what one would see with lettuce drop, these plants are firm. The roots do not just, it's not a soft rot, it's a firm root, but the plants do have some similarities to lettuce drop. Another thing that we're seeing more and more is confusion with Pythium. Pythium symptoms look a lot like this, and it's important to be able to differentiate these from one another. At least early on, the symptoms can look similar. The disease is associated with flooding or poorly drained soils, much like Pythium. Secondary factors that we found associated include plant stress and salinity, although we think those are largely secondary issues that are probably related more to the nature of the fields that are affected than the disease itself. Traditionally, this disease was believed to be caused by two Tombus viruses, Moroccan pepper virus, formerly known as lettuce necrotic stunt virus, and tomato bushy stunt virus. This is work done about 20 years ago by one of my colleagues when I was first hired here. It never quite matched 100%. As many of you know, we've for many years suspected there was a third related virus out there that we explained the other, say, 30 or 40% that we didn't pick up. And with the advent of high throughput technology, sequencing technology, we decided it was time to try to find this thing. And so that was what prompted the study. We began by working with folks out of UC Extension, predominantly Steve Koike initially, and a number of other growers in the area, requesting plants that appeared to have dieback-like symptoms. To try to get away from finding the same old every time, we tested these plants for the presence of TBSV and Moroccan pepper using degenerate primers as well as some specific primers, and selected the plants that came up negative. Basically, we were looking for anything that was different. With those, we then passes the sap from those infected lettuce plants or symptomatic lettuce plants to a series of other hosts that we use in virology to try to pull out viruses that may be sitting there and hiding. And through that, we found a really nice symptom on Nicotiana benthamiana, one of our more susceptible alternate host plants. In this case, the plants are kind of a bushy, yellowy pattern, as you see in the two photos on the left. And eventually they become more of a necrosis, which is fairly typical. These are very typical of what we see with Nicotiana benthamiana. And so based on that, we extracted total RNA from both the symptomatic lettuce plants and the test plants that were derived infected from the symptomatic lettuce plants. We then ran that RNA through high throughput sequencing. Initially, work was done at Boyce Thompson Institute by a colleague, which did not yield anything of interest, but then we repeated it with a private lab, and we're able to find some interesting results there, ultimately identifying a virus in a rather unknown family of plant viruses called the Phenui-Viridae that was not related to anything we were expecting, and not really related to anything any of the virology community knew. It looks like it's going to be its own genus. From that work, we identified 5 of 6 samples from the Salinas Valley and 1 from Yuma that were infected with this virus. We obtained the full genome of this virus. It, like I said, does look distinct. And although the virus is mechanically transmissible, it is poorly mechanically transmissible. It goes to Nicotiana benthamiana much easier than it goes to lettuce, although we have transmitted it to lettuce and repeated the symptoms, and I'll talk about that in a few moments. The nature of this is, it's really not that surprising. We've been working with this virus for years. If it was easy to transmit mechanically, we would have figured it out years ago. So I think it's the nature of what we're working with. Just to compare with the Tombus viruses, one reason we think we didn't see this before is because Tombus viruses, when they infect Nicotiana species, kill the plants fairly quickly. On the right, you can see Moroccan pepper virus on Nicotiana benthamiana. These plants don't get very big and they quickly die. In contrast, lettuce dieback associated virus creates a kind of a bushy pattern on the plant, and the plants do get a little bit bigger. So I think most of the time, if there was Moroccan pepper, we often would lose the plants early on. We just weren't able to see this thing. We correlated this with disease in the field. We continued to collect samples for a couple of years on this, and this just illustrates the dynamics. We saw virtually 100% correlation between suspected dieback symptoms in fields and the presence of lettuce dieback associated virus. You'll notice on here samples 1833 and 1836, which was a TVR1 resistant romaine variety, were both negative for symptoms and also negative for the virus. This is typical. We also tested a number of non-symptomatic lettuce plants with the intent of just seeing if this virus might be a cryptic virus that just hides out and we don't see it. We just accidentally discovered it. We did not find this virus at all in any non-symptomatic lettuce, which again suggests this is the real deal. We looked at our freezer samples that have been collected over the past 20 years with varying degrees, but the important thing is we don't know how stable this virus is, but overall we found a very high correlation with suspected lettuce dieback samples from the freezer and the presence of lettuce dieback associated virus. We believe this virus very likely is somewhat distantly related to INSV. It's in the same order, which is a very large grouping. So one of the things that we think may happen is it may not be super stable in the freezer. And this is one of the things we've seen with INSV as well. And I'll talk about that in a few minutes as well. We sequenced the genome of the virus. It has 3 RNAs. I'm not going to get into the details of this, but as a typical virus has, it has proteins associated with replication, movement, and protection of the nucleic acid or the nucleic capsid protein. Does appear to just have the 3 RNAs. In comparing it to other viruses that are known around the world, again, it looks like it's going to be its own genus. It is related to this watermelon crinkle leaf associated virus that was found exactly the same way with high throughput sequencing in China, and Laurel Lake virus, which was also identified, I believe, in the same manner. So these are viruses that I think are now being discovered because of this new technology. If you look way down the list, you'll see tomato spotted wilton and patient's necrotic spot under tospovirus. And again, it is the same order, but it's a very large grouping. And I would not suggest that these are going to behave particularly similarly, especially with regard to vectoring. I do not think this has an insect vector. So what does the genome tell us about lettuce dieback associated virus? It is, again, like I said, related distantly to INSV. Both are in the Bunia viralis, but it's in a different family, and it's also probably its own, like I said, its own genus. Most members of the Finui viridae, interestingly, are insect transmitted, which is, again, very interesting to us. But the nature of this virus, the way it behaves in the field, it does not look like it will be insect transmitted unless it were a soil-borne insect. That is something that we do want to look at more. Alejandro Del Pozo and I had hoped to do some of that work prior to his departure. But we actually think the more likely vector will be a soil-borne fungus-like organism, although we don't have that information yet. It's been a difficult year to kind of pull that together, but it is something we're going to be looking at. Finally, we do not anticipate seed transmission either with this virus. Another interesting aspect is we've now found the virus in both the Yuma area and the coastal areas. Coastal isolates, including Fresno County, are closely related to one another, basically 98 to 100% identical based on sequence comparisons of the replication proteins. Desert isolates are also highly related to one another, but they're only about 95% identical to one another if you compare across the areas. So this suggests some adaptation based on growing region. So just to kind of go over where we are with our 2020 objectives, we've done pretty well in spite of COVID. We're not quite where we want to be, but we've done pretty well anyway. We've basically confirmed the association of this virus with disease and lack of association without disease. We've demonstrated that we know the genome of this virus. We've determined its taxonomic relationships. And we're currently doing studies both with lettuce containing the TVR1 resistance gene, as well as susceptible lettuce, to look at infectivity and symptoms, with some success, but not as much as we would like. This is actually the main thing that has been slowing down this project at this point. And I think it's something we may have to use cloning to development of an infectious clone to really overcome well. And then we are interested in a biological vector, but there are some challenges to tackling that. That's going to be a long-term project. That's not something we're going to be able to do in a year or two. That's something that's going to require larger amounts of grant funding and a longer, more significant investment, probably through, say, a state or federal funds, which we have applied for, by the way. Where we are now in conclusively proving that lettuce dieback associated virus directly causes the disease. We have been propagating the virus in Nicotiana benthamiana. One of the things we learned in the past year is this virus does not freeze well. We've worked with frozen isolates. It is possible to transmit from a frozen stock, but it is not efficient at all. Now that we're beginning to see field samples this spring, we're working with live tissue again, and it is working so much better to get transmission. We believe that by freezing the material, we're losing some of the infectivity, which suggests to me that the virus may, and I don't know this yet, have a membranous outer, may have an outer membrane like some animal viruses have, much like we see with INSV, although we don't have proof of that yet. It's something I'd like to do, look at this under EM and see what we can find. We're now working with inoculating lettuce plants that have been adapted to make them more susceptible. One of the things we're doing is working with plants that have been kept in the dark for 24 hours prior to inoculation, because this does sometimes enhance infectivity. And then we're looking for symptoms and monitoring by PCR. This is just showing you how we do that. We inoculate very young benthamiana plants to establish infection from lettuce isolates collected from the field. As they progress, you can see the differences between plants that become infected and those that do not. The tall plants in the photo on the right are not infected. You'll see the one in the front appears to have some symptoms developing, and the one in the back on that row is also showing symptoms. The whole middle row is showing strong symptoms of the virus. And it takes some time. It takes about 3 weeks to see good symptoms on these, although you can sometimes see infection very early if you have a hot isolate. And we've got some that have been showing within 5 to 7 days. Other times you see it may take up to 3 weeks. This is an example of inoculation of lettuce. Along with that, we always inoculate the benthamiana because the infectivity is better on benthamiana. But we're looking at, in collaboration with Yvonne Simcoe, a number of TVR1 resistant lines as well, just to compare. And again, they're being inoculated very young because they're more susceptible. We're still not seeing great results with this, but we think that may be improving. Most of these pictures that you're seeing here were done within the last month. So we're very hopeful now that we're working with field isolates again, we can be much more efficient in our transmission tests. One of the other things I want to mention is, and this is what it looks like when you see infection on the left, you'll see a healthy plant versus a plant of the same age that became infected with LDAV. This was confirmed by PCR. And you can compare the symptoms to what you see in a field plant as in the one on the right.
Bill, you have 5 minutes.
I'm almost done.
Okay,
We're just
perfect.
wrapping up. So moving forward, what we want to do, and I'm going to do this in a little bit in reverse order. I'm going to mention objective 2. We're wanting to develop infectious clones of lettuce dieback associated virus. We believe this will enhance delivery of the virus to test plants using an agro-inoculation system. Sometimes this can work much better than traditional mechanical transmission. And so that's one of our goals this year. That will require in-lab time and is something we have not had a lot of with the rotating schedules that we've had during COVID, but we see that opening up a little more in the next few months as we begin to come out of the pandemic, at least we hope we will. The other piece of the puzzle is we believe it's very important, especially with diseases with similar appearances in the field, to have an antiserum that allows us to quickly say this is lettuce dieback associated virus or it's not, to differentiate from other diseases that look very similar to this. And I believe that's something we can do fairly quickly. Again, it will require lab time, doing some cloning, gene expression or protein expression, and development of antisera through a commercial company. We're beginning that work through this project. We also have a proposal to the California Department of Food and Ag's Specialty Crop Block Grant Program to continue this work on a larger scale beyond what we can expect through this project. This is actually the last year we plan to do this project, and we're going to be focusing a little more intently on INSV as that's become the big player in the area. I do think we can wrap this up and make it a nice little package for the companies to be able to develop tools to screen germplasm. One of the things that we've been lacking is a great system for inoculation in the greenhouse that'll allow rapid movement. Yvonne has some great markers for the TVR1 gene, and if we have a good assay system, I think things can move very quickly to get this in a lot larger number of varieties of lettuce than we've been able to do to date. And then finally, we do want to look for biological vector. I anticipate it will be a soil-borne organism like maybe Opidium or Polymyxa or something like that. Most likely Opidium is my leaning. Again, we do not know yet. It'll take some time. Those studies are very involved and will take some serious vetting of a vector to figure that out. I do not believe it'll be insect transmitted unless it's a soil-borne insect, but that's something we can also consider. It's clearly a soil-borne disease that likes high soil moisture situations. So with that, I'd just like to thank everybody involved, particularly Laura Jenkins-Ladke and Viviana Camello, who have worked extensively on this project, and Steve Koike, Richard Smith, and Bindu Pudel for their work in obtaining samples. We've worked closely with a couple of the companies and some PCAs. And I'd certainly like to acknowledge Yvonne and Dimitar Malev from USDA Beltsville, soon to be USDA Corvallis, who has assisted me with some of the high throughput sequencing work. And thank you, of course, to the program for continued support on this for the past few years. And again, this will be the last year of this project this coming year, but we will continue to transition this into larger types of state and federal grants to complete the work while we move in other directions on local virus issues. Any questions?
Okay, great. We do have a poll for your presentation, so that's up now. And once we complete the poll, we'll break, and let's still reconvene at 11. So quick break. And then I don't see any questions right now, Bill, but please just keep monitoring that Q&A. I'll email you too if I see something pop up.
Okay. And of course, if anyone has concerns about whether their situation in their field has dieback, of course, let us know or let Steve's group know. Steve's working with our diagnostic system. He's done a great job with it. And he's got the resources to look at that, Steve Quicky's group. So certainly, if you do have questions about it, Steve or I can either one assist you with that.
Great. Thanks, Bill. And then
Okay that I volunteered you, Steve.
it looks like people did pretty well on this poll. If you want to just quickly address it.
Yeah, so again, I threw out some virus names that many of you have heard. The viruses that are clearly associated but not probably causing disease are tomato bushy stunt and Moroccan pepper. Lettuce dieback associated virus is the virus that we've identified that appears to be the real causative agent that we've been characterizing. So yes, you all did really well on that. We do not know the vector yet for this. We suspect that it's a soil-borne organism. We do not know what it is yet. So most of you got that right as well.
Great. Yep. Thanks. All right. So let's take a break and we'll start back up at 11. So give you 4 minutes.
Thanks, everybody.
Thank you. Okay, it is 11 o'clock on my watch. So Steve Finnemore, I see you. If you want to share your screen.
I'm trying to get the, okay. I was trying to get the laser up. Okay.
Oh, I think once you hit presentation, now try
now
it. Now it's over in that corner.
Yeah.
Too.
Okay.
Perfect.
All right. Good. Thank you. So we'll continue on here talking about weed management, and I'll end by talking a little bit about STEAM and some disease management, which is quite relevant. It's interesting hearing about the Pythium. So I want to acknowledge the extensive work that Elizabeth and Richard Smith did on this in the in-field evaluations. As you all probably know, Richard Smith and I are not new, and both of us are older than Curb. And so it's great to have Elizabeth, a new weed scientist, contributing, and more than contributing. All right. So the objective was fairly simple, was to evaluate the weed control efficacy of these autonomous weeders. And so we did work with two units. The orange unit is the FarmWise machine, and the green unit is NIO from France. So they did, there were two types of evaluations here. The FarmWise machine is called Titan, that's the orange machine, and the Dino is the NIO machine. It's the green machine. So the evaluations, the data collected were weed control and hand weeding times, as well as lettuce yield. And so this is basically what it looks like in the field. On the left, you see the standard cultivation where you're leaving an untouched band so that you don't damage the lettuce. But the limitation is that it misses a lot of the area. Whereas with the intra-row cultivators, so they're cultivating between the lettuce, it's much like, basically, I think of it as a robotic hoe hand. And so, because it's doing a lot of the work that a hoe hand can do. And the idea here is the efficiency, of course, reduction in labor costs is really the objective. And so, again, the evaluations are weed control, and this was all done in lettuce, the time of hand weeding, and then the yield. And so there's a little bit on the statistics. There's Richard, and I'm not sure who all is there in the crew counting weeds. So a little bit on the results. So these are the percentage of weeds removed On the left, you have the auto weeders. So it's giving you it's removing about 75% of the weeds. So again, that's doing that mechanically. The standard cultivation is removing something like 35%, which is of course significantly different. And then for weeding time, the desired effect is to reduce that as much as possible. And the automated weeders did reduce the hand weeding time compared to the standard cultivation, but it was not significant. You'll see later on the research station, we got a little bit different result, and I'll explain why. And so here's the count of the lettuce stand, and basically we don't have any difference observed. So there was no reduction in stands, and then the lettuce head weight at harvest, there was no difference. So then we go to the field station. We did a little bit more elaborate, and that's the objective of doing the work on the field station. We had the dyno cultivator compared to a standard cultivator, and we had curb at 2.4 pints per acre compared to no curb with both cultivators. So it's called a 2x2 factorial. And so here's what we saw. This is weed control, the percent weed control. This is with the dyno, the blue. This is with curb. So we're getting about 80% weed control with the dyno plus curb. This is a much higher percentage, higher population of weeds than you would see out in the commercial field. We like it that way, so that we can detect these differences. And so there was a difference between the dyno and the standard cultivator in terms of weed removal, and we saw that trend whether we had curb or not. See, the next slide is showing the hand weeding time. So where we had curb, we naturally had better weed control. So we had lower weeding times, and there wasn't that much difference between them. That's similar to what they saw on the commercial fields. So this is the reason we do this is in the case where we left curb out, we of course knows curb contributes a lot to weed control. So the reason we did this is to see, to sort of unmask or peel back the effect of the herbicide and see what this cultivator is really doing. And it really is helping a lot because it's significantly reducing the hand weeding time. The overall hand weeding times are higher without the curb, but that of course is in conventional is not what we're doing. So these cultivators really are helping to reduce the hand weeding time. And so in terms of yield, there was no difference, which is the same thing that they saw in the commercial fields. So a summary, the autonomous weeders are removing significantly more weeds compared to the standard cultivation. There was no difference in the hand weeding on the commercial fields. There was where we left off curb. So you probably would see that in a organic field as well. You would where you don't have the effect of the herbicide. So there was a significant reduction in weeding time where we had high weed pressure, and that's what you would expect. And the good news is there was no effect on yield. So acknowledgement to the people that contributed to all of this, and to all the people that did the hard work. And thanks to the collaboration from FarmWise and NIO, as well, of course, as the growers that work with us. So a little bit about future work, and here I'm going to say a little bit about STEAM. So we're going to continue to evaluate these autonomous weeders because we're convinced that crews of these autonomous machines are going to probably replace the hand weeding crews that are getting harder and harder to staff and to pay for. We're also continuing to work on STEAM, and I think basically the idea here is we're using we're using water. Of course, we're converting it into steam, and we're actually able to kill quite a few pests. I'll show you what we're doing. We're working with a French company, that Cymox, that makes a really nice boiler that's easy to mount on a machine. The design you see here, we're working with an engineering shop down in Gonzales that's designed a platform. And what this is a four-wheel dolly with a bed shaper on it that's got hydraulics here, and you can't see the hydraulics here, to raise and lower this thing and basically inject steam into the soil on 40s and 80s. And we're trying to make it adaptable so we can also go on 42s and 84s down in Yuma. This is a collaborative project with the University of Arizona. They're excellent collaboration with those guys, and Mark Siemens and Barry Pryor, the plant pathologist at Tucson. Hopefully there'll be more on him. Basically, there's three things you can do with steam. The broadcast is where you treat the whole field. This is a machine we tested from Korea that does that. There's band steaming, which is what Mark developed here, came up with a design. Mark Siemens, that is, University of Arizona. And then spot, which we basically is a future plan where you actually put steam in like a 4-inch circle, and you would transplant, or you would drop a seed in the middle of that. This is going to be engineering wise quite a bit more difficult. So we're working on the band right now. So what's the deal with the band steaming? Well, the weed control is pretty good. Here's the result in a trial that we did last summer here in Salinas, where we had steam and a control. And so here's the difference in weed control with hairy nightshade, shepherd's purse. Little mallow is a little tougher. We also have trouble with little mallow and fumigants, burning nettle. So burning nettle and shepherd's purse and hairy nightshade are fairly susceptible. So what about diseases? I wish I would have put a Pythium slide in here too, because steam does very well on Pythium. We have a lot of data on that published and in recent results that we haven't published yet. But I put lettuce drop in here. Graduate student Ellie Guerra is working on this project. She pulled soil samples before and after steaming, so pre and post. So here's what steam did in the first trial. 5.6, it reduced it to 1.3 sclerotia per plate. Here's the control. There was no change. And basically you see the same trend in the second trial. So we can control sclerotinia minor. This is what it looked like. The band steam is on the left. The untreated control is on the right. This is a fairly modest pressure. Okay, Fusarium. This is Mark Siemens' data and Barry Pryor's data from Arizona. Mark's machine that I showed you earlier, it's the blue machine going 0.5 miles an hour. And 0.75 miles an hour compared to the control. They're getting pretty darn good control of the Fusarium in the lettuce there. So we're pretty excited about that. So what are our objectives to the Leafy Greens Board? We're going to evaluate the weed control efficacy of the autonomous weeders. We're going to try to combine that with steam in at least some studies, so that we can, the overall objective is to try to reduce hand weeding as close to the need for hand weeding close to zero as possible. It would be nice if a benefit, of course, is to control some of these soil-borne diseases, which seem to be an incredible challenge. So that's what we've got going. I also should mention that for the first time trying to work with the Carrot Board, because of course they struggle with Pythium a lot. That's the cavity spot. And so the STEAM project, we're trying to get some involvement, because of course, developing machines is expensive. So I'm trying to work with as many people as possible to come up with the best possible design for a machine. So that's what I've got, Jennifer.
All right, great. And I think we do have a poll question or 2 questions, I believe. There we go. We'll put that up. And let's see. I think something's gonna pop into the Q&A for you. Looks like Rami has a question. It's not all the way in yet. And I guess Steve, if you will, just monitor that Q&A in case some questions pop up. But it's nice to start off a section a little ahead of time, so we'll keep moving. Steve, Steve, I see a question came in. Let's go over that poll, and then I'll give you a minute too, or 30 seconds to answer that Q&A question.
Yeah. So the results, the amount you're going to reduce the weed densities is going to vary based on your, what the weeds are and how close they are. But 50% is about right. And then in terms of the reason to adopt the automated cultivators, basically, it really stuck with me when one grower told me, yeah, I like having hand crews doing, lettuce thinning, but I can't get them exactly when I need them. And so I need options. And so that really stuck with me because it totally makes sense. So that's what we've got.
Okay, great. Thank you. Everyone did well on that. And Rami's asking, how deep does steam control weed and disease with your research?
Well, the disease, I'll take the weed part to begin with. Basically, the weeds that are coming up in the top 2 inches. And so you don't need to go that deep to kill the weed seed. With the disease, that's something Mark and Siemens and Barry Pryor and I are trying to understand, and we've requested some federal funding to try to deal with that. But we think it's around 6 inches. If we can control for fusarium, if we can control a 4-inch wide band to about 6-inch depth, Barry Pryor's theory is that we are protecting the young lettuce plants from early infection with the fusarium. So that's probably the most formidable pest that we have. The good news is Pythium is pretty susceptible to steam. We've so far been able to kill it every time. That's my measure of, if we don't kill Pythium, we didn't do a good job. So.
Thanks, Steve. And with that, we'll get Michael Kahn.
Sure. Yeah, so I have 2 talks to present, or 1 talk, but 2 projects to present this morning. And one is about water treatment technology using polyacrylamide to control sediment in agricultural runoff, and also to help improve chlorine treatment of tailwater. Yeah, so I'll talk about this first project, which is on using polyacrylamide to control sediment and runoff. We often see this on the east side of Salinas Valley when we're running sprinkler systems that we get runoff, and it carries with it nutrients, sediments, and pesticides. And a lot of the pesticides are actually attached to the sediments. And with the current Ag Order 4.0, or that's going to be adopted in April, growers are going to be responsible to not cause toxicity for the runoff that's leaving the farm. And pesticides have different degrees to which they adhere to sediments versus be in the water column, in the liquid part of the water. And pyrethroids, which a lot of growers are, of course, transitioning to from the organophosphates, are very strongly bonded to sediments. So if those sediments are leaving in runoff, you're also pulling those pyrethroids with it. In addition, on the east side of the Salinas Valley, many growers traditionally have used retention ponds to capture the tailwater that comes off their ranch. They would be put at the bottom of the ranch and capture that water, and then it's reused for, say, germinating up a new crop. And so this was the traditional way of preventing that runoff going off the property. But because of food safety concerns, this practice has really been limited, and especially because the LGMA rules have very low thresholds for generic E. coli in that water that's being reused. And so in many cases, to get below the 10 colony forming units per 100 mils, growers are really needing to chlorinate that water. Now, if the water has a lot of sediment and organic matter in it, you need a lot more chlorine to get below that threshold. And so this is an opportunity also to use the polyacrylamide to drop out that sediment and make chlorine more effective. So polyacrylamide, or for short, we call it PAM, is historically been used for erosion control, and it can also be used to drop out suspended sediments and runoff. And these are special types of polyacrylamides that are just for soil erosion and approved for agricultural use. And they are very water soluble, although they don't often just go right into the water, and I'll talk more about this. They have large molecular weights, so very long polymer chain molecules. And they're the ones that are used for soil erosion are moderately anionic or negatively charged. And so they wrap around these suspended sediments in runoff and cause it to flocculate out, is how it works. So years ago, we did a lot of trials demonstrating the benefits of injecting these liquid versions of the polyacrylamides into the wellheads on commercial farms using this equipment. It's a metering pump, and it was very effective in dropping out suspended sediments if we got the water to have about 4 or 5 parts per million of PAM in it. But this equipment required calibration and some skilled labor to really operate it. Plus the equipment was expensive. And also the liquid PAMs, which are emulsified with mineral oil, are fairly pricey compared to using, say, dry products. So we want to revisit using PAM, but in this case, figuring out how we could use dry PAM and get it into these pressurized irrigation systems. So there's granular PAM available. There's also tablet forms available, and they're much less costly than the liquid formulations. The granular is about $2 a pound, and the liquid might be around $8 or more per pound. And also have a system where we really reduce the need for labor to operate the system. And then another benefit is that we found that the mineral oil in these liquid PAMs that we used years ago have toxicity, have aquatic toxicity, which really negates the whole point of using the PAM. So we could avoid this because we were just using straight PAM, which we know is not toxic to aquatic organisms. So the project objectives were to refine and scale up a prototype applicator to inject dry PAM product into overhead sprinkler systems that would use in commercial vegetable fields, and then evaluate the effectiveness in field trials. And then this third objective is to evaluate using the PAM for improving the efficacy of chlorinating tailwater intended for reuse in leafy greens. So this is a picture of the sort of finalized prototype of the PAM, dry PAM applicator. and you see it's a chamber, and on the right side, let's see here, right side here, you have a cartridge that we fill up with the dry PAM, and we insert it in there, and surrounding it is this static mixer that causes the water to interact with the PAM. So once the PAM is wet, it becomes this gooey material. It is water soluble, but unless you get that water to really stir around this, you can't get it very easily into the water. So we did some investigations on how to get this to efficiently go into the water. Let's see if I can show this. I have to get rid of the laser pointer, it looks like. So here's the first 4. It's a static mixer with 4 mixing elements. Oops. Not that one, this one here. And so you just see it's filling up, and we put some bubbles in the water so you can see how it moves and turns around the PAM cartridge here. And then we went to 8 mixing element static mixer. And so you get a lot more turbulence around that cartridge and get the PAM more efficiently into solution. So this is the finalized system, and we're doing field tests here. And so we can have 6 chambers that are in parallel, and we can treat 180 to 200 gallons per minute through this system. And we have a drop of pressure about 6 psi when we do that. And this is not how you would use it, in the field. It's really designed to be at the wellhead, and so you just like a filter station, you would push the water through it. But for field testing, we actually set it up in the field. And so we ran so far 2 field trials, and we intend to do a 3rd one, and maybe a 4th before this project is completely over. And we would do the test during the germination phase of the crop. These were done on the east side on Chular loam soils, and there was approximately 5 inches in the first trial applied and 6 inches of water in the second trial. And so we had these plots where we could monitor the water coming off as runoff and measure the volume, as well as take samples throughout the irrigation to evaluate how much sediment was suspended in that water. And so these are the results of 2 trials, and you can see in the flume here, the water is much clearer compared to the untreated plots. And we had 92% reduction in turbidity by using this system. And in terms of suspended solids, about 86% reduction. We also saw that we also reduced the amount of runoff. We had more infiltration of the irrigation water, and it averaged out as a 43% reduction in runoff. And so when you multiply the change in runoff volume times the concentration of sediment suspended in that runoff, we were getting a 92% reduction in sediment loss from those fields. So this translates on average, to about 70 pounds of sediment if it was untreated per acre lost in those 6 irrigations versus just 5. And just to sort of scale that up, if this was over 400 cropped acres, we'd potentially reduce the sediment loss from 14 tons to less than 1 ton. So the other objective we had in this project was to evaluate using PAM for improving the efficacy of chlorinating tailwater that would be intended for reuse. And you can see all the sediment that the tailwater has in it. what we want to do is introduce PAM into the water going into those sediment ponds, those retention ponds. And the way we did this was we adopted this auger system for applying PAM into a canal, but in this case for runoff. And so we had this component called a stilling well with a float switch that would turn the system on proportional to the flow rate going through this weir. And then we had automated sampling pump upstream and downstream to measure the effect of the PAM. And so you can see downstream about, I would say 400 feet or so, the water is very clear. And so we are reducing that sediment more than 95% in the runoff. And so you have very clear water going into those ponds. And we'll be evaluating what the effect is in chlorine needs requirements are by having that sediment reduced. But this is sort of a summary from our data from the summer and the winter. And the blue is upstream turbidity concentrations or turbidity units versus downstream. And you can see we went from over 2,400 turbidity units down to 43, which is really in the range of what the water board wants growers to get to if water is going off their property. So it was a 98% reduction in turbidity by using this equipment. So to summarize this project, the dry PAM applicator, that we were using to getting that PAM into the pressurized system greatly reduced the suspended sediments and presumably also the pesticides that would be associated with those sediments in the runoff. And it also was able to increase infiltration, which means you could operate your irrigation system for a shorter period of time and get the same amount of water into the soil. The dry PAM applicator requires very minimal labor to operate. And the concentration of PAM that's going into the water is actually lower than in the previous method using liquid PAMs. So we're estimating that at this point, probably the cost of the PAM is somewhere around $5 per acre per crop. The auger applicator, shows a lot of potential to greatly reduce the suspended sediments in the tailwater retention basins. And so our plan is to continue with some more field evaluations, and we want to scale up that dry PAM applicator and try to get it on a wellhead. And we have a grower that's willing to cooperate on this, so we could really evaluate it on a larger number of acres, and then continue to evaluate the effect of PAM on the efficacy of treating tailwater with chlorine in 2021. So now I want to just change gears and talk about the second project, which is using single-use drip tape to germinate up lettuce, which is now a pretty common practice in the Salinas Valley. And one of the advantages of doing this is that you eliminate the need for sprinklers. And if you're fighting wind issues, you can eliminate wind patterns as well as surface runoff. And for crusting soils, you avoid that soil crusting effect. And because you're using single-use tape, you're using new tape every time. And so you have a very high application uniformity. And there's less labor associated with using the tape for removal. And because it's new tape, you don't have as many repairs. And most of you are aware there's some specialized equipment for saving labor and extracting the tape from the field. Because you're not reusing the tape, it can get recycled and you start off with new tape. And so there's no labor required for doing the tape repairs. So really the objective of this project was to compare water and nitrogen management and weed control of lettuce under buried drip using the single-use drip with the traditional overhead sprinklers for germinating up a crop in commercial fields. And then we also wanted to evaluate the effect of irrigation water volume on lettuce seedling emergence using berry drip, as well as nitrate leaching. So we were fortunate to have a lot of cooperating growers that were willing to split their fields where half the field was germinated by drip and half by sprinklers. And we would put flow meters out there in each half of the field to monitor how much water was being applied and the pattern of irrigation. So this is a summary of 7 field trials that we did last year. The blue being the drip managed versus the sprinkler germinated areas. And to sort of our surprise, you know, we did not see less water being used for drip germination compared to sprinklers. In fact, on average, a little bit more, about 6 inches versus 5. Now, it seemed to be one of the effects was the soil type as well as the management and who was the farm manager. But where we had silty clay or clay loam textured soils, it was often challenge for those growers to get that water out to the seed line from the drip tape that was buried in the middle of the bed. And that's because those soils are often very well aggregated, and the water doesn't just move laterally right across very rapidly. And so it would take quite a bit of water. And another factor was that growers want to assure that, you know, all areas of the field are getting sufficient water. So if you have a problematic area in the field, it takes more water. But, you know, with the sandier soil types of textures, generally the growers were able to use a lot less water for germination, you know, using the drip, but they also tended to use less water even with the sprinklers. So there was also a management effect out there. And another factor we noticed was just the uniformity of these irrigation systems and how they were being managed. So I'll talk more about that later. So
Michael, you have 5 minutes.
okay, very good. So the germination rates were similar between sprinkler and drip germinated. That wasn't too surprising because growers are definitely trying to get maximum germination in these fields. And the soil nitrate distribution, which we evaluated after the germination, tended to be similar for drip and sprinklers. We did have concern that drip might cause more leaching of nitrate because all the water is being applied in the center of the bed versus more dispersed with sprinklers, but that wasn't the case. And then we looked at weed control. And one concern is that drip germination may not activate some of the herbicides like herb that are being applied, or it might actually prevent curb from leaching. And in these cases, we didn't see any differences in weed control. So because we did notice growers were tending to over irrigate in some fields, we wanted to work with a grower that had a sort of a track record of using a lot of water for drip germination and see if we could utilize less water and still get a good germination. And so he put us on a fairly challenging field, and we created a replicated trial in his field using these lay flats. So we had the grower standard, we had a low water treatment and sort of a medium water treatment out there. And what we found was we could reduce from the grower standard of about 13 inches of water down to as low as 4 or 5 inches of water and get similar germination. And this is a croply, silty clay soil, and you see how well aggregated it is. And often it looks like the water's not moving out to the seed line. But what you have to really do is look below the surface and make sure that seed is moist. Now, for fairness for the grower, he had very long runs with his drip system, and they were running it at a very low pressure. And so at the end of the fields, that water wasn't moving out to the seed line. And for that reason, just to get that water at the end of the field to uniformly germinate that crop, you had to run that water a lot longer, which brings us to sort of, what we're interested in doing this year is to work with growers and sort of demonstrate and evaluate if we can improve how that drip system is operated and run, and make some changes in the design. Can we use a lot less water for the drip germination? So what we've learned so far with the fields that we've surveyed is that germination rates, water use, nitrate losses, and weed emergence was actually similar among drip and sprinkler germination. But the potential water savings using drip for germination is probably going to be dependent on the soil type, irrigation system, and the management. So what we're planning to do is evaluate some additional fields in 2021 and investigate if we can improve the pressure management and the uniformity of these drip systems. Can we use the water much more efficiently for drip germination of lettuce? So I just want to acknowledge Leafy Green Research Board for sponsoring this project and all the contributors to these projects. And I want to mention also, we've worked very closely with Mike Ray at RayFab in developing the PAM applicator, and one of the reasons, or the thought process, is that growers are interested in using this PAM applicator. Mike will be able to construct it for them. So I'd be happy to answer any questions.
Hey, Michael, we think we do. Oh, we do have a poll for you. Sorry.
Yeah, and it's 3 questions, so make sure you scroll down.
Thanks. So we'll give that let's give that some time. And then Michael, you do have a couple of questions in the Q&A if you want to just take a look at those. And then we can answer those once we finish the poll.
Okay. Yes, the one question is, does PAM break down over time? Yes, the PAM will break down under UV light. It's just a very weak polymer. And the question is polyacrylamide toxic? The ones that we use for agriculture, for controlling soil erosion, are not toxic. And I participated in a study on this, and we got the water up to 100 parts per million, and it still had no effect on the water species, the water bug species of interest. I think was the Hyela azteca, which is the sort of a shrimp-like little critter in the water. Although what polyacrylamide does is it makes the water more viscous. And so when you get to 100 parts per million, it's pretty hard for that little bug to get around in the water, but it did not cause any toxicity. Does PAM increase food safety concerns? The concentration we're using with our PAM applicator is probably less than 1 part per million. And in terms of using it on crops, these PAMs are all registered with the Department of Food and Ag as safe to use on food crops. So if you're going to use a PAM product, make sure they have that label. But yeah, it's completely safe to use.
And,
so,
yeah.
Yes.
Mike, I was just going to comment about the food safety. I mean, I think, right, the best use of PAM is sort of early on in that crop's life, right? And as you get closer to harvest, maybe you're not using PAM on the crop. Would that be
Well-
accurate or no?
If you're going to continue to sprinkler irrigate, right, then you'd probably use it. But most growers in the Salinas area certainly are switching to drip for their leafy greens. But then you have leafy greens, the high density leafy greens that are going to be continually irrigated, with overhead sprinklers. So you could use it there. Again, I guess I would emphasize, talk with your buyers and make sure it's, that's on the list of safe products to apply before you use it close to harvest. But in terms of what the Department of Food and Ag is concerned about, that it is safe to use on food crops. It is a pretty commonly used product in food processing. So for example, they use it for clarifying juices because it can flocculate out, the solids in juices. It's also used in makeup, so it's a pretty common product for a lot of different uses.
Thanks, Michael.
Yeah.
And then, yeah, if you want to address these questions, since I got to see them beforehand, I don't know that everyone did so well on number 3.
Oh, okay. Irrigation runoff can transport which of the following to downstream water bodies? And most people got this right, both sediments, pesticides, and nutrients. And then which class of pesticides is most likely to be bound to sediments and runoff? And a lot of people said all of the above, but the best answer would be the pyrethroid pesticides because they have the highest KOC. They tend to want to be stuck to sediments. Now, all of the pesticides will stick. All these classes will stick to some extent to sediments, but only the pyrethroids would be mostly on the sediments. And then the last question, which class of pesticides can polyacrylamide help mitigate the most in runoff? Well, that would be the pyrethroids because the PAM is going to drop out the suspended sediments. And so the pyrethroids would be attached to those suspended sediments. So I hope that makes sense.
Yep. And for those of you PCAs that will need to take the test, I hope you made a note. And so with that, Michael, thank you. And Richard Smith is our next presenter. So Richard.
Okay, well, good morning, everyone. And what I'm going to talk about is this, get this thing in the way, the development of nitrogen uptake coefficients for lettuce. And this project is motivated by what's coming down very shortly, by the way, with regards to the Ag Order 4.0. And in this Ag Order, which is the basically regulations that are coming down on water quality, they're going to use an A-R target or limit. And A being the amount of nitrogen that's applied in the fertilizer that's applied to the crop. And R is what's removed by the crop. And in the original Ag Order, that was kind of the way it was written. And so what we're trying to do then is understand what is removed by the lettuce crop. And that you would think that was something that we might have a handle on, but it turns out we didn't really know it very well. So on the A side of the equation, when you talk about what's applied, well, it includes the fertilizer, obviously, right? But then they were also including the amount of nitrogen that was applied in the compost. And then if you were in organic production, they were also looking at the A that's in the applied, that's in the organic fertilizer. and then also the water, what the nitrogen that's applied in the water. And so Mike Kahn and I have been working with the Regional Water Quality Control Board and other people, giving them feedback. And so in this revised order that's currently under consideration, they made some modifications. And so there is some good news. So, there's no getting around the A that's in the fertilizer, but they had a different way of evaluating how much nitrogen to credit from the compost. And so they made, based on input that they were getting, they adjusted, the amount of nitrogen, a credit. So based on the carbon nitrogen ratio, they adjusted it to either a 1/10 or a 0.05 factor. So they're not going to count all the nitrogen that's in the compost. That's good. That was very important. And then for the organic production, they're using a regression. I'll show you that here from Patricia Lazicki's paper to calculate how much nitrogen credit they're going to give, how much of that nitrogen that's applied in the fertilizer is counted as an applied amount, because not all of it becomes available. A lot of it becomes, becomes part of the organic matter, actually. And then in the irrigation water, Mike Kahn made a very great improvement, helping them understand that we just count the amount of water that the crop takes up rather than all of the water that's applied. And that's a big factor that's really going to help growers be able to comply with these regulations. And then here's just this regression that Patricia had in her paper. And it basically shows you, like some of these granular fertilizers, maybe only 50% of the nitrogen that's actually in the fertilizer becomes available. And then that's what the growers are going to be, that'll go into their A factor. Now, when we talk about the R side of the equation, this is a little more complicated, but not really. I'll just explain it. Of course, there's the R that's in the harvested crop. And that's mostly what we're going to talk about today. But then they also have the sequestered. This is like in perennial crops, which of course not of concern to us in vegetables. But then they have an R scavenge, and that's what's in cover crops and in high carbon compost. And this is something that we're currently working with the board on, and hope to make some progress in getting some modification, because the way it's currently written, there's not much of an incentive to growers that are using cover crop or high carbon compost. And then there's an R treat, that's what's removed in denitrification beds that we heard about earlier this morning. And then R other, in my mind, there's still some questions that need to be answered here, because of denitrification on certain soil types, certain irrigation systems actually can be a little more significant than we might realize. So that's something that needs more research. But this is what we're up against with these A-R. They start off as targets, but then in 2027, they become limits. And so this is the total amount. A-R currently is, it's going to be, starts at 500, but then, and starting in 2025, it starts ratcheting down, and then ultimately becomes, some numbers that are quite low for, double crop vegetable crops. These are numbers that we need to come to grips with, whether we could ever, fully meet these limits, but that's to be determined. But what the Regional Water Quality Control Board is, they wanted to know, okay, what's the crop coefficients for all these different crops, 95% of the acreage in the Region 3. And then here's a list of crops that actually this project that the Leafy Greens is funding is helping us address. And this is a list of crops that not just Mike and I are dealing with, but also Aparna in Santa Clara and Andre and Ventura counties are working on to try and develop crop coefficients. But for this project, we're obviously concerned with lettuce. And so in 2020, we worked with this group of lettuce products and worked collecting samples. We coordinated with growers and then collected samples. We had a few hiccups with the COVID issues, but that all got worked out very quickly. And so we collect the samples directly from the crews. We do 6 reps per field. They're immediately, we measure the moisture content, and then dry them and then remeasure them to get the percent moisture. And then we send them off to the lab up at Davis to get the percent nitrogen content. And then what you see is what's interesting is that, and this is something that this project's really not set up to address, but there's actually quite a significant amount of variability, even just in the percent moisture that's in a crop. So we might see something like for full-size romaine, the percent, this is percent, not moisture, but percent solids. So it might, vary from 4.6 to 7.5, which is quite high, and with a mean, of course. And then nitrogen might vary. So, this all obviously gets back to variety, irrigation strategies, and fertilization and so forth. And but then we take this number, and then we, based on these numbers, then we develop this coefficient. And then you can take that number and you can multiply it against what's your net yield. So let's say this romaine weighed 30,000 pounds coming off of that field, which is interesting to me because I'm talking to growers. Sometimes they know how many boxes come off a field, and then what's the net weight of those boxes. That's always something a little bit not as clear cut. And so that's something that the growers are probably going to have to, in dealing with these regulations, understand better. I'd like to understand it better myself too. But you can see if you just have a number, which is I think what the regional board wants, a coefficient number, but the reality is that it actually varies quite significantly. And okay. This is the problem. And I just want to talk about this because it's something, this gets to the heart of the problem. Okay, so like, let's say this romaine crop, 54 pounds of nitrogen was taken away in the boxes, down the road, out of the field. That's your R factor. But the reality is, if we look at how much nitrogen are people putting on, this is numbers. This is growers' numbers from 2017 delivered to the water board. And then they plotted it out based on how much nitrogen, the quantity of nitrogen, and then the number of ranches, reporting using these different numbers. The shaded area is actual nitrogen uptake by the crop. And you can see in general, there was a mean of about 175 pounds of nitrogen applied. And the problem is, if you're only taking 54 pounds of the 175 away, then you got this quantity of load of nitrogen that remains in the field. And, we've estimated those numbers, but now we're going to have more precise numbers. It can be quite a significant amount. And then if you do that again, so let's say, you have 100 pounds left over, A minus R, and you do that twice, see, then all of a sudden you start to add up, you start to bump into these limits. And so what Mike and I have done different scenarios looking at, well, what's achievable by the industry currently. And what we see is that, okay, maybe making the 500 or the 400 limits or targets, that might be doable, no problem, but once we start ratcheting down to 300 pounds, or 200 pounds, then it becomes quite a more significant issue for the growers to to comply. And so, when you look at this A minus R, I call it an imbalance, and so what can we do on the A side of that? Well, this is where all the good practices that we've been talking about for all these years with Tim Hartz and others, using the nitrate testing and efficient irrigation. Mike's been preaching that, and he's got, Crop Manage to help people do it. I think nitrogen technologies could be a bigger factor, down the line. The nitrification inhibitors, nitropyrin is going to be registered on lettuce. So some of these technologies could be helpful. The other thing, though, I think that's important is on that R side. One of the things that the board in the current revised draft order, they were only giving 30 pounds of credit to cover crops and to high carbon compost. And one of the points that we want to make, because there is one more chance to give public input to this process. You have to sign up by April 9th, by the way. You go to the website and you can give comments. And then the comments period will be, I think it's like the 14th, 15th, and 16th of April. But anyway, cover crops, this is something that we've worked on over the years and seen how much nitrogen they take up. That's kind of a bridging practice, so like if you use a cover crop, it sequesters whatever quantity of nitrogen that's in it, gets it through the rainy period, and then puts it into the next year. And that's good. And so how much credit should it get? Should it be 30 pounds as is proposed? Or, what we've observed over the years is cover crops take up, 100, 150, even up to 200 pounds of nitrogen. I make the argument that should be the credit, because that's what it's capturing and reducing in terms of nitrate leaching during that winter period. And so we'll see it, if we can make that argument. High carbon compost, that's kind of an ongoing project. But we've seen that also can sequester a certain amount of nitrogen and get it through the winter period and get it into the next spring, at which point then it starts to break down, and then you can account for it with your nitrate testing and so forth. There might be other demonstrable forms of nitrogen removal, and that's where this denitrification, not all of it's leaching, some of it's going off as a gas, and we need to understand that, but that's going to take a whole another project. And I already mentioned this cover crop credit and the high carbon compost. So that's where I will stop, but I do want to acknowledge Trish, who's just been working so hard on this project, and Tom Lockhart for helping us, and then the student helpers over the summer, Carmen said, and then the funding from the Leafy Greens Board. We, of course, greatly appreciate that. And then we also got funding from the FREP program. So that's it, Jennifer.
Great, thanks, Richard. I don't see any questions right now, and this presentation does not have a poll question. So with that, I guess let's keep monitoring the Q&A and see if some questions arise.
Yeah, I'll keep an eye on it.
Okay, thank you. And
Thank you.
then Ali Montazar is our next presenter. And I see his screen. Oh, there you are, Ali. I didn't see you. Now I see you.
Yeah. Thank you, Jennifer.
All right, thanks.
Yeah. Good afternoon, everyone. I'm very pleased to be here today. So my talk is about drip irrigation in spinach. As you might know, we initiated this project in 2018 along with Michael Kahn and Alex Putman. So far we conducted 4 field trials in our research center in Holtfield, mainly on organic baby spinach. We also worked with the commercial fields, actually 3 commercial fields belong to one of our growers in Imperial Valley who adopted drip irrigation for conventional bunch spinach. Even though we didn't do a specific trial, but we worked with him on economic analysis and also some measurements for yield, irrigation efficiency, and practices. So it was a learning curve for all of us. We tried an experiment and treatment. We eliminated next time, we added a new treatment, or we continue based on what we learned. So what we did in year 1 and year 2, we evaluated 2 drip depth, soil surface drip and 1.5 inch depth drip, 3 and 4 drip lines per bed, 8-inch bed, 2 nitrogen regimes, and germination by 4 drip line per bed. So we learned some from that, that experiment. But in year 3, we evaluated 2 planting methods and germination by 3 drip lines per bed. We also did some changes on nitrogen strategy. We basically applied some top dress nitrogen before first irrigation as a new practice. So since this is the last year of this project, I need to walk through what we learned from the past trials and then present what we learned this year. So what we learned from the first 2 years, so we didn't see a significant yield difference between surface drip and subsurface drip with the same drip line. But we also learned surface drip is not practical. So it would pose a food safety risk and risk at harvest time. And until the plant is not well developed and fully developed, wind causes some issues with this practice. But we observed significant yield difference between sprinkler irrigation and each of the drip treatment in trial, or first trial and third trial, but no significant yield difference between sprinkler and 4 drip line treatment. We also didn't see a significant yield difference as a result of higher rate nitrogen application for drip treatment, which is specifically about injection fertilizer. We learned downy mildew incidence is lower in a plot irrigated by drip. This is one of the solid results we received so far. And the local grower who adopted drip, he's very satisfied with this technology for conventional bunch spinach. So this is what we learned from the earlier trial. So we left the first 2 years with some thoughts and concerns about nitrogen management, about germination spinach seed by drip, and of course about economic feasibility of using drip in spinach. So we tried with the new trial to find some solution or some thoughts about these issues. So as I mentioned, the fertilizer application is the same before. We use True 662 as pre-plant fertilizer and True 413 as complementary fertilizer through injection into the system. But the difference between this trial and the other trial was we applied 40 pounds of nitrogen per acre as top dress before first irrigation. That's the only difference between this trial and the other trial. And we went only with 3 drip lines per bed in this treatment. So we don't have any 4 drip lines. So the other difference between this trial and the other trial was we also evaluated a strip planting method, which the grower, following this practice for bunch spinach, for conventional spinach. So we tried to evaluate this strip planting method also with a standard planting method. So 3 million seeds per acre used for a standard planting method and 2.8 million seeds per acre. This is what we use for a strip planting method. So in terms of germination evaluation, this is the result we received. So Here is the number of plants per 2 feet of the length of beds. So you obviously see there is no significant difference between plant population of a sprinkler and most of drip germinated plots. But the mean number of plants per 2 feet acre was slightly lower in drip irrigated treatment, 2.3 points percent less in drip irrigated plots, which is not that significant. One of the other solid conclusions we have, and we learned about this actually in all 4 trials, it's about crop canopy development. So what we learned shows the leaf density of drip irrigated treatments was always slightly behind of a sprinkler, between 2 to 5 days behind, which means we have a longer season, about 2 to 5 days longer season for spinach in drip compared with sprinkler irrigation. So in terms of total nitrogen content of plant tissue, this is the result I have for 35 days after first irrigation. So you see there is no nitrogen deficiency in both drip and sprinkler irrigated plots. Even in that specific days, so the mean total nitrogen content was slightly more in drip irrigated plots compared with sprinkler irrigation. This is a very interesting result. So you can obviously see in the pictures, the standard planting and strip planting on that day. You don't see any issues in terms of nitrogen deficiency or issues. So please just look at the picture I have in the right side of this slide. It's a close look in part of a drip irrigated beds. You see how uniform and high density we produced spinach, baby spinach in drip irrigated beds. I think I'm very satisfied with this trial this year. So in this trial, we try to learn about I think, yes, this is something wrong with my slide here, about the numbers, the days after first irrigation. So we measured the yield every 3-4 days after 37 days after planting. The result is very interesting. So as I mentioned, so the drip irrigated plots are behind. So the yield we have for, it means, the yield we have for 51 days after planting for drip is very close to 47 days, days after planting in sprinkler irrigation. So which means, it's about 1 to 6% less, still is 1 to 6% less, the yield in 50 days after planting in drip compared to 47 days after planting. But if it compares the yield of drip 51 days after planting versus 47 days after planting with a sprinkler, this is the yield we have. You don't see a significant yield difference among the sprinkler treatment and most drip treatment. So the mean yield we observe in bunch spinach commercial field is very close to the drip irrigation we have. But as I mentioned, that's bunch spinach, that's 8-week crop. So the baby spinach is about 6-week crop. So, but the yield is very close because the density, plant density in bunch spinach is lower than baby spinach. So as I mentioned, we had 2 planting methods. So this is the result we have. We observed an average of 2% yield difference between the planting methods in sprinkler irrigated plots, but 4% difference between the planting method in drip irrigated plots. When you compare this with the differences of the seeding rate between planting method, I think the result is very promising here. So one of the other solid conclusion we have is about downy mildew incidence, which in all these trials we observed downy mildew incidence while the disease pressure was not generally, high in this trial, but the downy mildew incidence numerically was lower in drip irrigated trials, 2 to 5 times less in drip irrigated class, which is I think is a good result here. So the only difference between these two irrigation practice, drip versus sprinkler, is the leaf wetness, I believe here. So the leaf wetness comes from dew from irrigation and rain. If you eliminate the hours we had dew and rain, so we can find out what is the difference between the irrigation time and the wetness are coming from irrigation. So for this 20-day period for this specific trial, so we observed 22% less, time of the canopy was wet in drip irrigated. But the observation we have for trial in the past shows this number might change, might vary between 22 to 26%. So we, economic feasibility is the main question probably everyone has, but it's not an easy task to do, evaluation on this, because we need more data, we need, more experiment on this. But we have some data from a grower here. We have some data from our trial, and since I'm working with Imperial County Farm Bureau to do some economic feasibility assessment for the water management we have and water conservation practices we have in the desert. So I have some numbers, and I came up with a very simple economic analysis to give you some idea how feasible might be drip irrigation in spinach. This is, by now, and all the experience and the data we have by today. So this is the numbers you see here that are the annual cost difference between drip and sprinkler. So of course, we have higher costs as a result of drip tape and inserting and removal drip tape costs. Totally about $800 more cost for drip irrigation compared to a sprinkler. So what we learned from growers, the fertilizer cost will be about $120 to $150 less, because we can conserve fertilizer in conventional. We don't have any evidence about organic, even in this trial we did. So we didn't reduce the organic fertilizer application, which means we don't have any data to see how might affect reducing fertilizer in organic. But the worst scenario is we don't conserve any fertilizer in organic. Let's assume this. So in terms of water, if we go with germination, with drip, we cannot conserve water. Mike Kahn just mentioned, even in our trial in a research center, we applied about 4% more for germination in drip compared to a sprinkler. So which means we might, not able to conserve water. We didn't even improve the yield production, not growers, not us in this trial, but the yield data are very close. But the grower reduced the energy cost by $200 per acre. The labor cost, it's very site specific. It really depends, if the grower has a pumping station or they have portable pump, that's different scenario and a story. But about $150 to $250 per acre, we might be able to conserve a labor cost. But the game changer here is the lower cost for downy mildew control and water treatment for food safety. So what we learned from growers, $300 per acre less cost for these treatments. So if you just do a simple map here, you see drip could have $100 per acre less cost to $100 per acre more cost compared with sprinkler irrigation. This is not a wide range, specifically because we are talking about adopting a new technology in a new commodity. All the experience we have in the past, when we start a new technology, you know, there are a lot of optimization ahead. You know, if we start adaptation with this narrow, you know, values, I believe the technology is feasible for spinach in the future. This is the
Well, you
conclusion.
have 5 minutes.
Sure, thank you. I'm almost done. So which means our evaluation based on you know, everything we have by today. So my evaluation is economically is feasible, but of course management makes a difference here. So what we learn shows we can germinate spinach by drip. As I mentioned, just about 2% to 3% less germination rate by sprinkler, which is good. And we are talking about very high density, you know, baby spinach, 3 million seeds per acre. So 3 drip lines per 80-inch bed could work with the soil type we have in the desert region, you know, with the silty clay loam or clay loam could work. That's why, you know, we considered this trial with 80-inch bed and 3 drip lines. So the strip planting method has a better performance regarding nitrogen related issue. We didn't see any specific issue, but still we, you know, in some spot we saw with the standard planting in our trial some yellowing issue and nitrogen deficiency. So there is no evidence until now on yield improvement and water conservation for using drip in spinach. As I mentioned, we should expect a longer period to be harvest, you know, spinach field in drip. We significantly might reduce downy mildew disease. You know, if the disease we have a high disease issue in the region, and of course reduce food safety risk. I think that that's the main game changer here. So again, as I mentioned, so you know, economic feasibility is very site specific, but you know what we learned here, it shows economically drip irrigation for spinach is feasible, specifically for bunch conventional. You know, we are very confident on this, and of course, based on the data we have, it could work for baby conventional spinach in the future. Again, management makes a big difference here. So with that, you know, what we are doing from now, we are publishing the results of this, you know, this project in the next future. We will continue doing outreach activities. You know, I did a lot of outreach, you know, a lot of interview and some extension publication. I'm hoping in the future we see some more spinach field in drip, hopefully. With that, I'm going to offer my special thanks to California Leafy Green Research Board for all the support you provided in this period, you know, the last 3 years. The support we received from Imperial County Agricultural Benefit Program and several cooperative growers. Not so I didn't work with just one grower, you know, several growers. They provided some thoughts and information to this project. I'm thankful to all these individual and organization. Thank you very much.
Thank you, Ali. So we've got a couple of poll questions up for this presentation. And there is one question in the chat from Michael Kahn, and he was wondering if you saw less yield with the strip planting.
Yes, and the reason, you know, as I mentioned, so the seeding rate was less in strip planting, and we saw a little bit, you know, less yield here.
Okay, so let's
exactly right. That's exactly right. Even you know, with the strip planting, with the sprinkler, we observe less yield in.
All right, Ali. Well, it looks like everyone got 100% on the first.
Yes, I think, you know, this is what actually we learned from this project, so we can reduce downy mildew significantly. And the question 2, again, the majority got it right. That's you know, what is likely the main reason that we see less downy mildew incidence in drip is less leaf wetness.
Great, thank you. And then Ali, just also watch the Q&A and see if there's any other
Sure.
questions that come up. And
Sure.
then with that, our last presenter is Daniel Hasagawa, and he gets a fair amount of time because he's got, what are we doing, Daniel? 3 projects, 3 projects, 3.
Yeah, so I think I have 30 minutes, is that right?
Yeah.
12:55. I think we should be done by then. So anyways, thanks to everyone for sticking around, hanging in there. I've really enjoyed all of the presentations so far. And so you should be able to see my full screen presentation. So
Yep.
3 projects that I'll be talking about, they've all been affected in some way or another, and we won't spend too much time talking about that, but mostly focusing on some of the outcomes and what we have been able to achieve over this really challenging year. So I'll walk you through these. The first project is our area-wide monitoring project for aphids on the Central Coast. And this project was initially led by Alejandro Del Pozo in collaboration with myself here at USDA, and then Ian Grettenberger at UC Davis. So once he departed early summer, we decided to revise this proposal and really think about what is actually achievable for this funding cycle. And so we trimmed that down to really the first 2 objectives is, continuing some of our monitoring and then possibly improving some of the reporting system that comes along with it, just to get that information to the industry, to growers and PCAs a little more efficiently. And then the second objective here was really an extension of what we had done in 2019. So monitoring populations for this emerging new aphid biotype that affects Central Coast lettuce. So the first thing that just to talk a little bit about the trapping that we've established and how we're reporting it. I think a lot of you have heard this already, but we're using double-sided yellow sticky cards. We run 21 traps throughout the valley from Castroville to King City. We replace them on a weekly basis, and we break those down into averages based on the location. So Castroville, Salinas, Chular, Gonzales, Soledad, Greenfield, and King City, and then we also do averages. And so we're providing these updates mainly through the UCC entomology website, just because I know there's a lot of subscribers out there, so they can get that information readily once it's updated. Also to the listserv as well, I believe. So from 2020, keeping it consistent in terms of how we're breaking it down by cities, by general locations. We have weekly data all the way from January 6th up until the end of the year. We're continuing that into this year. You can see the most recent trends for March 23rd. And this information is quite valuable for PCAs and people in the field, just understanding what the overall movement and populations of insects are in their respective areas. Okay. And then we also provide historical data. So looking at trends over time, and I think this is really important to build this historical data set, not only for aphids, but other insects as well, such as thrips, diamondback moth, other major pests in our area. So we continue to monitor this. We're providing updates on a weekly basis, and we hope to see this continue into the new year, just to keep supporting the industry on some of these reports. I mentioned that a lot of this data is being posted to the UCC Monterey County website. So thanks to Richard Smith for helping me get this set up, as well as the administration there. It took a couple months to kind of work things through, but I think we have a pretty good system there. So you can see the aphid monitoring, thrips monitoring, and we also run traps for diamondback moth, and those are pheromone traps. I know that's not related to lettuce, but that data is being updated on a weekly basis as well. So the second part of really this project, the second objective of project 1 is looking at the emergence of the lettuce aphid, Nasinovia rebus nigri. And so first reports of some of these new biotypes present in the Salinas Valley in 2018. And the idea is that these new biotypes, they're morphologically indistinguishable. And so you can't determine, whether it's here or not just by looking at it. So you actually have to run resistance bioassays. But it was first reported in 2018 because it was able to infest a lot of these commercial cultivars that are planted that should have resistance to them. And so there was concern. We did some studies in 2019. And then the continuation of that was just to do some more surveying in 2020. Just from 2019, we developed these little cup bioassays, these leaf disc bioassays, where we take different populations of aphids and then we put them onto these leaf discs that either are susceptible or should be resistant. And what we saw was that from a Gonzalez population, we had some other populations that we tested as well, but from the Gonzalez population in this isolated area, we did see performance of both the nymphs as well as the adults that seemed to outperform the colony that we had at USDA. So that did suggest that the NR1 population was actually present in 2019. When we continued that, we were able to squeeze in 2 sample periods here. We're testing them on 2 commercial varieties here. So Holbrook, which is susceptible, Baranda, which is resistant. We didn't see any differences. And so really, what 2020 has taught us from this study alone is that we need to do some more extensive sampling. I think if you talk to a lot of PCAs and growers, especially organics that are utilizing a lot of these cultivars that have the resistance, they'll say that they're having issues with it. So that is highly suggestive that it is. Our data here just isn't showing that. So it's not saying it's not here. We just didn't capture it in this study. Okay. So in summary, we had 2 objectives really from this first project. And so we were able to establish and really continue the Salinas Valley monitoring, but we also updated that. So we're now doing reports on a weekly basis. So it's more or less real time for the industry, not only on aphids, but also thrips and diamondback moth. And then we were able to get in a couple studies with field populations for setting up bioassays, addressing the NR1 status here in the Salinas Valley. Okay. I'll just save questions until the end, and I'm just going to jump right into the second objective here. And this is really going to be the bulk of my presentation because one, a lot of this was done in the field. And so the restrictions in terms of COVID didn't really impact this project as much as it did, particularly for our third project. But and the other thing is that this has really become an emerging issue. And so we really wanted to prioritize this project here. And so one objective, but there's a lot of kind of deliverables and outcomes that we really wanted to achieve by the end of this project. And so the basic overview of it is let's expand the host range of INSV in the Salinas Valley. Let's understand what are the roles of cultivated crops as well as non-cultivated crops and assess their status as potential reservoirs for the virus. Let's do that over the course of the season. Let's do it in the non-season, non-lettuce growing season, and let's do it in different regions to really try to get a grasp on what's happening and to capture some of this really information, which can have pretty strong implications in terms of weed management. Okay, and so I've shown this before to several groups, and I don't think this is anything new, but if we look at a general map of the Salinas Valley on where INSV showed up, it was really in between Salinas and Gonzales, which was our heaviest areas that experienced INSV incidents. We had cases also up in San Benito County, up in Santa Cruz County, but all the way down to Greenfield. And I didn't take spotted wilt out here because I'll circle back to it at the end of this project, but we do occasionally see another thrips transmitted virus, which is tomato spotted wilt virus. And I think that's come up in some of the breeding talks earlier. Richard mentioned it in terms of gaining insight into what's known about resistance for spotted wilt. So I'm going to just leave that here as well, because it is another tospo virus transmitted by the Western flower thrips. So using that, and we actually had, I had a general idea of what these hotspots were going to look like in 2020. So that's how we designed this back in winter 2019. And so we had 4 sampling sites, this north area, which is around the Spreckles area, Salinas, a little bit in the Blanco area. We had the west side, which is kind of near the river, the foothills between, and then between 101, the east area, which was probably our heaviest hit area for INSV. And that's just based on personal observations. I don't have any strong data to support that, but just from all the scouting that we did in 2019, these are some of the reasons why we selected these sites. And then south was kind of an outlier in terms of this study, because there was less INSV incidents down there, again, from my observations. And what the approach was, we had these different sampling sites within these broad geographic regions that we would go back and sample consistently from these different time points. And so we chose these time points because we felt like these were very critical times in terms of when we're seeing transitions of lettuce production, but also transitions in thrips vector populations. And so majority of the lettuce here in the valley is grown from March to November. We know they start planting right after the lettuce free period, but this is a very broad calendar based schedule that we're showing here. So we started this project in winter 2019 with a broadcast approach. Let's sample as much as we can and just start testing it for the virus to see, to build this evidence. Are these plant hosts potential reservoirs for the virus, particularly during these winter months? So are they serving as these green bridges for the virus to propagate and then move back into the spring production cycle? And so 4 strategic sampling sites here. And that was the general approach. We tested materials using triple antibody sandwich ELISA, and then we tried to validate as much as we can using either immunostrips or RT-PCR. So the approach here moving forward, I'm going to talk about the broad sampling and then the 3 sample periods, really just the unique outcomes from each of those. And then at the end of my talk, I'm going to talk about top 10 hosts and how those different weeds fluctuate over the different sampling periods. So bear with me here. So this was our broadcast sampling. I'm not going to spend a lot of time here other than just to say that the darker the red color is, the higher the virus titer is within those samples. We have number of samples and then percentages. All I'm showing to you here is that we sampled 42 different hosts, 42 different species, and we're really starting to build this database of which weeds might be within our top 10. Okay. And so by the end of that, we were able to narrow it down. And this has been something that has really changed throughout the year. And as we continue to sample and understand which of these are better reservoirs, this top 10 list has changed, I think 2 or 3 times since we started this project. And so that's why it's necessary for us to continue doing this and really try to understand the biology and how these weeds are playing a role here. So, but this is the most updated list that we have. Pictures provided by Richard Smith here. So thanks for his help. If we break these down just by family categories, and then Richard and Elizabeth helped me on this one because I could not wrap my head around it, but we talk about seasonality and abundance of these weeds here on the Central Coast. If you look at the ANR website, the abundance of these weeds don't necessarily hold true as to what's typically described in California because of the uniqueness of the climate here. And so they helped me put together this nice little table, just very simple, talking about when these weeds are present. And you can see a lot of them are present year round here on the Central Coast. So spring sampling, the objective here was, again, I'm going to focus on the big impact from the spring sampling. And what we saw, what we wanted to do was look at, whether some of these weeds are actually serving as reservoirs from the winter into the springtime. And so what I'm showing you are just top 10, the top 10 at that time here. So evidence for winter bridge hosts. During the spring, we sampled a number of weeds. We saw that they had infection. But the other thing that we did in the springtime was we sampled with Richard and Elizabeth. Their support has been incredible here, is sampling weeds that we know were present in the wintertime and then sampling new flush that was coming up in the springtime. And what you're seeing here in the west side and then the east side is that we're getting positives in both the old growth as well as new plants that are coming up, suggesting that there is actually movement, but also this potential for bridge crops to actually serve as reservoirs for the virus when lettuce is not present here. Summer sampling, these are again, some new potential hosts that we described in the summertime. A lot of the cultivated hosts here, we specifically wanted to sample during the summertime, particularly when INSV was particularly bad in lettuce crops. And the strategy here with the cultivated hosts were that we went out and sampled the spinach, peas, artichoke, all these other crops in areas that were adjacent to lettuce fields that had INSV. And you can see really, spinach 4 out of 10 was a good host. We saw one pea plant that had the virus, but others we weren't able to detect it there. Okay. And then finally, this last time during the winter period, we tried to narrow that in as much as we could with the lettuce-free period to really address, is the virus here when lettuce is not in the valley? And so outcomes from that, we sampled 952 plants over the course of 2 weeks during that lettuce-free period. And I'm going to talk about the outcomes for the winter, but again, these are just new hosts that we're describing here. And chickweed was an interesting one. It's really one of the few that we've seen that shows strong symptoms of the virus. And what we've seen is that chickweed is primarily localized in certain regions in the valley, particularly the farther north you go in those cooler conditions. And a lot of this growth of weeds occurs in areas that cannot be managed by lettuce growers. And so it creates a really challenging dynamic in terms of thinking about weed management. In the south, we also decided to test for spotted wilt. We did see 2 out of 16 sow thistle plants test for spotted wilt. The grand total was 2 out of 118. And the reason why we wanted to do that is because really Soledad was the one location where we saw spotted wilt. Sow thistle is a previously described host for spotted wilt, so we weren't too surprised. But again, this is sort of a proactive type of approach to try to understand some of the virus dynamics, thrips dynamics, potential viruses that might be in our valley here. Okay, so let's see, I'm going to speed this up a little bit. So talking about the host range, in total, these are all of the hosts that we sampled over where the sample size was greater than 30. The average ELISA values are recorded here on the left-hand side. So the higher the value, the greater the virus titer is within that plant. And you can see lettuce is really good. And that comes from a plant that shows very strong symptoms. Same thing with chickweed. We've validated using immunostrips and RT-PCR for a number of these hosts that are very common in here. That provides really confirmation that yes, the virus is actually there. So one of the challenges that we had was, how do we rank these and how do we determine a top 10 list? And so talking to Richard and Elizabeth 2 weeks ago, one of the things we thought of was developing a ranking system. And so this is what I came up with. It's a host INSV index where you take the ELISA value. And again, this is the reason why we do ELISA and not just immunostrips, is it because it provides that extra layer in terms of understanding its relevance as a host. So ELISA value times basically the percent positive. So the number positive, if you look at chickweed, 26 divided by the total 50, so it would be multiplied by 0.52, which comes out to be 52%. And so the index value that comes from that are these numbers here. And that's how we sorted that. And I think that's really interesting to note, because you look at bull mallow, short pod mustard, very low percentages in terms of infection. And so when we apply this index, they're really low on this list, close to ice plant, close to some of these other things that we've tested that don't appear to be really good hosts for the virus. And so moving forward, I think this is one strategy in terms of prioritizing which hosts are actually relevant. In red, I'm showing you the top 10 list. This also just kind of draws attention to some of these other hosts, curly dock, ox tongue, pigweed, some of these other ones that we might have to revisit and really do some more extensive sampling to understand its relevance. I didn't notice that initially, and it wasn't until we did this indexing that really drew our attention to it. So one of the questions that comes from this that I want to talk about briefly is, just because it's a host for the virus, is it a reproductive host for the thrips vector? And so keep in mind that in order for transmission to occur to a plant, only the adults can transmit the virus, and it cannot be passed down into the next generation. And so each generation has to reacquire the virus specifically at the larval stage. And so what that means is that the female has to lay her eggs in an infected host. And so that host has to be a reproductive host for the thrips vector. From that top 10 list, from all of the plants that I showed, that's the big question, is how many of these are actually reproductive hosts? I think a lot of the broadleafs are, but we just don't know enough about the reproductive biology to know whether there are preferences in terms of hosts that are available in the Salinas Valley. So those are kind of long-term studies, but I just wanted to bring that to your attention, because just because it's infected with the virus doesn't mean it's significant in terms of providing that virus inoculum to the thrips vector, which is the only way lettuce can be infected. Okay, big pictures here, summarizing all of this data. So in total, we had over 3,000 samples that were collected for this entire project. We saw an infection rate of 13%. If you narrow it down to the top 10 hosts, those percentages are a lot higher for the top 10 hosts. And so I think we're doing a pretty reasonable job in terms of saying these are the hosts that are the most relevant, not only for their abundance, but also for their percent positivity. This is broken down by season: winter, spring, summer, winter again. What's interesting is when we look at, the total number, what I'm showing you are the least squared means here for a total number of the top 10s and their positivity value here. We see this general trend of high from winter 2019 to low in the summer, and then high again in the winter time. It's interesting. There's a lot of interpretations to that, and I have some ideas about it, and I'd be happy to talk to anyone about that, at another time. But, there's a lot of different ways that you can look at it, and I'd be happy to talk to anyone after this presentation if you have some thoughts. But certainly, I think what this is suggesting that, in the wintertime, we do have a high percentage of weeds that we're capturing that are reservoiring the virus. Okay. If we break it down by location, so north, east, west, south, I talked about this strategy of sampling and some of the reasons why we do that. Again, the same thing, we see higher percentages generally across all locations. However, when you break that down, some of my personal observations in terms of the incidence in lettuce, what we're seeing is that's actually sort of translating into the incidence of what we're seeing in the weeds as well. I certainly did not know that going into this, and the observations in lettuce are strictly observations, whereas this is some quantitative data that actually suggests that data coming out of the south, that those host plants are actually, less are not as infected as some of these other areas where we tend to see greater incidence of the virus. So again, another interesting piece of data. And by setting it up in the way that we did, it's allowed us to really draw these interesting conclusions and think about management from a systems-wide perspective. When we look at the relationship between season and location, and I'm wrapping this up too, we see this general trend, again, high to low to high, and we see interactions between season and location. So just as important are weeds that are not host for the virus, or at least we haven't been able to find it. So we've been talking about the top 10 hosts. We're now talking about those that we didn't find the virus, that are very relevant to the industry in terms of, surrounding areas, etc. So summary, I pretty much talked about all of that, so I won't spend much time here. Finally, RNA interference. This project is not moving as fast as I would certainly like it to, but that's really because of COVID, and being in a shared lab space with Bill has been fantastic, but you can imagine that having so many people in the lab vying for space during COVID has been challenging to make a lot of progress on this. So, but I will talk about some of the outcomes that we have. Again, these are a new class of biopesticides that are being developed by numerous groups across the world, and we're seeing greater traction, particularly application of RNAi technologies for pest management. So here it is, entomology right here in purple. So basically it's a nucleic acid that you're delivering, and it's been deemed as this species specific insecticide. We had done a lot of work prior to this position that I was in whiteflies in East Africa and the Southeast tomato production. And so what we're trying to do here is adapt them for thrips, viruses, and some other insect pests as well. But the idea is that you're applying these molecules that are specifically inhibiting the translation process within the central dogma that all of us possess, right? It's this natural regulatory process. The idea is that if you develop something that is specific to one gene and you apply it, you can inhibit protein translation for that specific gene.
Daniel, you have just under 5 minutes.
Awesome. This will be quick, Jennifer. Thanks. So RNAi value is specificity, and there's a number of papers that have talked about this, and I talked about this at the mid-year meeting, so I won't spend too much time here. But there's a lot of different methods in how you can apply it, different pathosystems that you can implement it as well. The biggest challenge for all of this has been coming from regulatory. How do you regulate these new types of molecules? And so I think a lot of the wrinkles there are being flattened out. And so we're going to start seeing some of these technologies coming to market. And hopefully this research here will be a little bit ahead of the curve in terms of trying to adapt it to our local systems here. So objectives was really very first phase of this: synthesize triggers, identify genetic targets, and start to do efficacy trials. And so basically we design sequences, 200 to 500 nucleotides. We look at specificity to either thrips or to the virus. In this case, we're looking at it to the INSV genome. We can also multiplex those to other viruses as well for better management that way. We synthesize these things in the lab, and then we conduct bioassays. So again, the idea is if we're targeting the NS, the small genome here, specifically this one gene, the N gene, the idea is that you can prevent protein formation of that and then effectively stop really the persistence of this virus within a host. So the idea is that we've been able to synthesize 3 Western flower thrips targets, 2 INSV targets. We have a number of them in the pipeline. We're starting to set up bioassays, so allowing plants to take them up into the root system and then really looking at efficacy, whether they're reducing virus infection and thrips infestation and feeding. So just a broad overview on where we stand. We're developing root drench assays. For this one target, we've been able to detect it in leaves for up to 14 days, potentially longer. And moving forward, we're really excited about setting up some of these bioassays. We have these different thrips feeding cages that we've established, whether it's on a leaf, on a whole plant, or whether it's in a leaf disc. And so in summary, this is a work in progress. There's a lot of entomology projects that I'm excited about moving into 2021 and 2022. And certainly I'm not, leading all of these, but I'm fortunate enough to be a part of many of them that really touches on both cultural strategies and looking at, optimizing resources that we have. And that's working with people at UC Davis, at Riverside, Imperial County, looking at, improving management and monitoring strategies, not only here, but also in the desert. So again, I'm really lucky to work with all the other people that are listed on this slide. And in the big picture, what I talked about for thrips and INSV, this is what we're working towards, is building this IPM model and hopefully this time next year, I'll be able to have some updates in terms of how we integrate some of these other objectives and outcomes into this working model. And so that's why I'm excited about this year. It's going to be challenging, but I think there's a lot of opportunity. So thanks for all of your support. Thank you to California Leafy Greens and the board for your tremendous help and support, especially the growers and PCA. So thank you.
Thanks, Daniel. And I believe we must have a poll. There it is.
Yeah.
And so we'll let people get going on that. And you do have a few questions I see coming into Q&A and a couple in the chat. And I just want to say real quick while people are doing the poll, sorry, Daniel, I'm just want to acknowledge not just, you know, the new researchers we have at CSUMB that I mentioned earlier. I do want to include Nathan Jew in that because I really love that bioreactor project. But also Daniel at USDA, and there's a few other new research scientists at USDA as well that are great. It's just kind of an exciting time to have this new cohort of researchers available to us. And now we've got Apurba Barman too at the Cooperative Extension in Imperial. He's an IPM advisor. So that's great. So yeah, next year is going to be a good year. Okay, Daniel, I'll let you to it. If you want to address the poll first.
Yes. So the poll, which of the following weeds are described host for INSV? And that's correct. 98% of you got it. So that's great. Yeah. Which of the following is true? 90% of you said all of these are true. Is that management of weeds during the off season is important. RNAi can be used in a lot of different applications. And then we also need to do more work to really understand the prevalence of these new biotypes of aphids. So great job, everyone. A's on both of those questions.
Okay, and now let's see if you want to hit maybe the Q&A first.
Yeah, Q&A. So Alex Putman. So hey, Alex, by expand the host range of INSV, I think you mean expand our knowledge about the host range of INSV. That is absolutely true. Yeah. So minor detail there. So thanks. Yeah, expand our knowledge of the host range. So INSV again, and that really has come out of it originated as a horticultural virus, but it's emerging and becoming more important in vegetable crops. And so, you know, our understanding of the host range is really starting to develop and improve the more we look at it. So thanks, Alex, for that clarification. Next question, did you survey riparian shrubs such as willows and cottonwoods? We did survey the riparian habitats. Willows, I think we did test. I don't think that was a host. Cottonwoods, I don't remember. There's a list of 76 species that we can look at, but I think 0 out of 100 something samples that we tested from riparian habitats were all negative, and that includes the large arundo plants as well. So what is your east range for sampling for Cindy Wells? So that the east ranged from, that was kind of between Chular Gonzalez, and then we've also gone all the way up to kind of Williams Road. Yeah. Western flower thrips, one and only vector, absolutely. So in our area, Western flower thrips is the primary vector. There's also the European flower thrips, and then also the, the tobacco thrips are 2 other vectors that have been described to transmit INSV, but we don't have any indications that those vectors are present here in California, or at least in our area. Okay, should I jump to the chat now?
Yep.
Let's see. So where does it start?
Let's see.
Richard Mitchell Moore. Have the NR0, NR1 been sequenced here and for Europe? So no, they haven't been sequenced. That's something I think would be really cool. They've been identified in Europe. NR1 has been identified in Europe. It's also been identified in Australia, I think in 2019. So that's would be a great approach to it. One of the challenges with aphids, though, is that sometimes, a lot of times, these different biotypes and physiological behaviors, those don't translate down to the genetic level. A lot of those are just how the genes are expressed in terms of adaptation. And so that's one thing that I try to think about in terms of, a genetic sequencing project too. But I think you're right on in terms of taking an approach to understand it. Yeah.
Showing my ignorance, what's the typical genome size of an aphid,
I don't know for lettuce aphid. For pea aphid, that is, I want to say that is about 400 megabases. I think so. I think it is.
Yeah, that's pretty doable.
Yeah. Let me know if you have other questions, Richard. Let's see. Ali, oh no, wait, Ali's.
So I think too, I might be at the top of it. So Daniel, is there any indication that greenhouses allow for survival of thrips and serve as a source of thrips or INSV later?
Yeah, that's a great question, Yvonne. Greenhouse production is certainly at the center of a lot of these concerns. And we don't know. I know there are specific flower industries that actually suffer from INSV infection that has an economic impact on them. Again, I said INSV is typically a horticultural virus that is now being experienced in vegetable crops. And so the flower industry is also really relevant in terms of not only how they're suffering from some of these losses, but also potential sources. What we know through our monitoring data is that in the valley, though, thrips are active throughout the whole year. Although they're lower suppression, lower populations in the wintertime, they are moving around. Whereas as you head further north or in the northeast, you tend to see these things where they're either hunkered down in greenhouses or there's a big migration at the beginning of the season. So I think, Yvonne, yes, they can certainly play a role in all of it. So Michael.
Michael Kahn, yep.
Yeah, Michael Kahn, what is the mechanism of the dsRNA to provide viral resistance? Is it coding for a protein that protects the plant? So the idea is that the dsRNA is actually specific to the virus genome. And so what happens is as you get binding to that, to the virus, you're actually triggering, this degradation process that is innate to either thrips or the host plant that you can basically reduce replication or potential for the virus to replicate in the host plant. So you're not actually introducing new proteins or anything like that through the RNAi mechanism. Let's see, Apurba, do the weeds show some INSV symptoms too? Did you select the symptomatic class? Yeah, so that was one of the things that's important to note for chickweed, because that was symptomatic. That's why you see those percentages a lot higher, is because there was some biasm that was introduced when we were sampling. And so we were, same thing for lettuce, right? Those percentages are going to be your likelihood of testing a positive plant is going to be a lot higher if you're sampling symptomatic plants. So, but chickweed was the only one that I can remember that showed really obvious symptoms. Good question, Apurba.
And then at some point I'm going to cut it off, but there's
yeah,
a question
let me
about
know.
artichokes and if they are considered a symptomatic host.
Artichokes, yeah. So we've done 30 samples of artichokes, and I think we've only had one positive that was barely positive. And I haven't seen any symptoms in artichokes. If you do have a plant that is positive, I would love to see it and love to test it. So.
And then do you want to speculate why there's less INSV lettuce infection in South County?
I don't know, Rami. I mean, so one of the things that we thought going into it was maybe there's just less thrips down in South County, but that's not true. Our monitoring data suggests that the thrips populations are sometimes even higher in South County than North County. So it's not vector pressure per se. In terms of weeds, there isn't anything obvious that I see in terms of populations of weeds. So it's not like we're seeing little mallow, in all these other areas and it's absent in South County. We're seeing pretty good distribution of a lot of these down there. So the reservoirs are there. Why we don't see the virus in the weeds as much, I don't really know. I think a lot of the movement and the epidemiology of the virus certainly is driven by the vector, which I think is also driven by movement through the wind. And so I think there's a lot of environmental things, whether it's temperature, wind movement, that is probably playing a role in all of this that we just haven't even touched yet. Sorry, don't have a great answer for that.
Thanks, Daniel. And I just went ahead and put your contact information into the chat for everybody. So you can,
Okay,
you could
great.
tell me if that was terrible offline.
I
But
don't know.
I know you're good at, I know you're good at taking calls and emails.
Yeah,
This is a hot topic.
did you give him my I didn't give him my personal cell phone.
No, just your email address.
No, I think that's totally fine.
Your
Yeah.
home,
No,
your
I'm-
home
I'm-
address so they
yeah,
can drive
right.
to your house.
Right, exactly.
So anyway, real quick, I know we're running a little long, but I would be totally remiss if I didn't quickly, and Kelly, can I share my screen? I just want to acknowledge Jim McCrate, and he couldn't be here today because he's doing what retired people do and having a wonderful time in his retirement. But Jim did, work with the board for 44 years. And so this plaque will be sent to him since we're not meeting in person. And I know he wanted to extend his thanks and appreciation to the board for all of the support the board's given him during those last 44 years. So anyway, with that, I just wanted everyone to realize how much we appreciate Jim and the work that he's done. And not just research, but getting USDA's new labs, all that building up and going too. So anyway, I don't want to take everyone's afternoon. I know I'm hungry, but I just wanted to acknowledge Jim. And so, okay, Kelly, you've got the screen back. And I think Kelly just has a couple wrap-ups things.
Does it look fine?
Yep, that looks great.
Great. So there is the Certified Crop Advisor QR code. You can open the app and take a picture on your phone, and that should give you credit automatically, no problem. If you don't have the app or you have no idea what I'm talking about, you can just send an email to anrprogramsupport@ucanr.edu with your full name and your CCA number, and I'll get you signed up. For DPR, yesterday you should have already received your exam. Today you'll get an exam. I'm going to take a lunch and then send it right after. So you got to take the exam and pass with 70%. You have 1 week to take the exam, and I'm kind of a stickler with that because I have to get the information to DPR. So 1 week, and once I close it, I can't open it back up. Results, including certificates, will be sent within 1 week of you taking an exam. And again, 1 user per device. If you need anything or any questions, I'm going to keep this open for a little bit, and I'll be on to answer messages in chat.